topic, so which is given to me. So I will discuss my this lecture in two sections. One, I will discuss how we have investigated the carbapenem resistance mechanism. In the second part, I will discuss how we have tried to find out some solution for this carbapenem resistant bacteria. So first, we have collected the different clinical strain from all India Institute of Medical Sciences, and we have seen that what is the pattern of this antibiotic resistance. And we have found that if you see all the collected strain, this ATCC is not clinical strain, or there is a clinical strain. We have found that either the combination of antibiotic or carbapenem is going to show some sensitivity to clinical strain. So you can clearly see that. So either we have to use carbapenem or combination of antibiotic to control these pathogens. But at the same time, we will also see that there is a rise of resistance against those carbapenem and other combination of antibiotic also. Because here, if you see that, they all are resistant. But here, some sensitivity is there, but there is a rise of the resistance. Because my aim is to investigate the carbapenem resistance, so I have determined the MIC for the one of the carbapenem, that is imipenem, and we have determined the MIC, and we have found that around 35% of the collected strain are very high resistant to the imipenem. It clearly shows that it is going to be a very alarming sign for use of this carbapenem antibiotic. So because as I already told you that this bacteria have the membrane, and that the membrane is basically an interface between your antibiotic in the blood and then your pathogen. So we have seen that when we have treated the sensitive strain with the imipenem, we can see that there is an clear cleavage, you can say the destruction of the membrane. But if you see the resistant strain, it is going to have some changes in the membrane, but it's not going to basically completely lyse this bacterium. So it clearly shows that there might be role of this membrane, or there may be some changes in the membrane or some other biochemical features in this particular bacterium, so that it is going to be the resistant. So we have started investigating it. So what we have done is we have developed a protocol to isolate the bacterial membrane. So we, as I already discussed in the introduction, we have firstly separate total membrane of the bacteria, and then we have developed protocol so that we can easily separate outer membrane protein from inner membrane fraction. So in inner membrane fraction, we have this inner membrane protein and periplasmic space, and in outer membrane fraction, we have only outer membrane. Then we have taken any of either outer or inner membrane fraction, and from the resistant bacteria and sensitive bacteria, and then we have labeled these protein with the different fluorescence dyes. For example, we have taken resistant protein from resistant bacteria and labeled with the Psi3, and protein from the sensitive bacteria and labeled with the Psi5, and then we have performed the dyes analysis, and we have also performed replicate of this and then dye swapping also to neutralize the effect of one dye. And once we have find out this label protein and run the dyes analysis, we have analyzed the result by two independent software to neutralize the biasness, and we have found that two software decider identified 19 upregulated protein and 4 downregulated protein. Progenesis identified 14 upregulated and 4 downregulated protein. So if you see that there is a change in the fold difference, but we have selected the protein which is basically identified as overexpressed or downexpressed in both the software. So we have taken out those protein spots, performed trypsin digestion, and perform LCMS analysis, and identified by this mascot software. And what we found, you can see here in inner membrane, these because we have developed protocol which is having some contamination, 10% same with the outer membrane fraction with some contamination of inner membrane. So because it is not difficult, it's very difficult to separate totally 100% with 100% efficiency. So if you see that this beta-lactam antibiotic like carbapenem enter here and bind to the penicillin binding protein, but in the resistant strain, what will happen? They have change in this channel OMPW. They basically alter this penicillin binding protein. They have overexpressed OXA51 beta lactamase. They have overexpressed MC beta lactamases. They have overexpressed various proteins that is involved in the metabolism as well as in the protein folding. So it clearly shows that there are a role of these protein in the resistant strain, which might giving the resistance to the resistant strain. But as you know that proteomics is usually explaining the overexpression and down expression, it clearly not showing is there is a direct role or not. So we have further analyzed 5 protein of it like PVP, AMC, OXA51, and then Seprons, and then this OMPW. But I'm explaining this OXA 51, where we got a very good result. We have basically, as I told you, that this OXA 51 we have further characterized. So because you know OXA 51 is a beta lactamase, and it is known that if you give nitrosamine, then this beta lactamases cleave this nitrosamine into from yellow to pink color. So you can see that we have taken this all the fraction of protein fraction of this bacterium, and then let this perform isoelectric focusing, and then they let this yellow color substrate nitrosamine, and you can see that pink color. So this clearly shows that the presence of the any beta lactamases, but it may be OXA or maybe other beta lactamases. Then we have taken out these spots and perform LCMS analysis, and we found that these three these protein spots are the OXA 51. Then we have taken this OXA 51. So we have cloned this OXA 51, and then we have taken sensitive stain treated with the imipenem. As you know that this sensitive stain is going to die. But when we have transformed this OXA 51, you can see there is a changes in the membrane morphology, or you can say there is a survival of this bacteria enhances. It clearly shows that this OXA 51 have role in the carbapenem resistance. At the same time, because we have cloned it, so we have purified this OXA 51 and performed some biochemical assay, and we have found that they are going to cleave this nitrosamine, and they are going to cleave this imipenem. So this clearly shows that what we have OXA 51 we have purified is functionally active, and we have characterized it, determined the kinetic parameter of it, and we have found that it's going to have Km, Kcat, and Kcat by Km, which is very good in terms of their substrate specificity. At the same time, because we have purified protein, so we wanted to see that is there any effect of the because this acenato is going to survive at different harsh condition also. So we wanted to see that what will happen if you change this the normal condition of this OXA 51 to adverse condition. So we have changed the temperature, we have changed the pH, and we have found that the secondary structure of this OXA 51 is very stable if you change the pH, but its tertiary structure is going to change when you change the pH. So at the same time, when we perform the isothermal calorimetry, we have also found that this OXA 51 is going to hydrolyze this carbapenem in an exothermic process, which clearly shows that they are going to be a very good substrate. This imipenem is going to be very good substrate for the OXA 51. Then, because at that time we don't have the crystal structure of OXA 51, so we have modeled it, and we have performed the docking by two independent software, Glide and Gold, and we have seen that this OXA 51 has a very good binding affinity with the this imipenem, which is a carbapenem, and it clearly shows that when we perform MDNS analysis, it shows that the glutamic acid 111 is a very important residue involved in the binding of this OXA 51 with imipenem. So this is a one story we have just briefly explained here. But the second story, we have, if you remember, I told you that we have separated outer membrane from inner membrane. So we have performed proteomics of outer membrane also, and what we have found, you can see here. So we found that this overexpress specific transporters, overexpress efflux pump, we have found that overexpress iron binding protein and altered the caro. So I will explain how, because this I already told you that proteomics is not a technique which which will let you know exactly role of the protein, but it will say that it is overexpressed. But so we have to validate it. So we have further validated it, but so I will explain one by one. So one more thing that because when you are giving if you see that host pathogen interaction, so the iron play very important role. So we have also done one proteomics where we have created three conditions. In one condition, we are two condition. In one condition, we are giving excessive of iron, that is FeCl3, and in other condition, we are giving excessive of this iron chelators, that is BP. So basically, we have created iron deficient condition and iron rich condition, and what we have found that, so if you create iron deficiency in Acinetobacter baumannii, for Acinetobacter baumannii, there is overexpression of free transporters, ferric acinetone vector receptor, ferric enterochelatin receptor, and there are different receptor which is overexpressed that is meant for the iron binding. So it clearly shows that the bacteria is having different mechanism to proteomics result at least shows that they have different mechanism
I think there is some problem we are facing. There is some problem at Dr. Vishnath Tiwari's end. Dr. Tiwari?
I think
I think you got disconnected, Dr. Tiwari.
yes, maybe
Yeah,
because the
please.
lab net is gone, so I've connected with mobile now.
Yeah,
So
and
should I continue? So
yeah,
where we
please.
left,
But we
maybe
have,
we just one slide before, I think.
yeah.
Where we left, sir? I'm sorry. So,
No, I think not. This is not the slide where
so this you have
you
seen
should. No, you were talking about this.
iron.
Yeah. Next,
Okay,
next,
so
next.
what we have find. So we have
Yeah.
found some iron responsive protein also. So maybe I will quickly move to the next one. So now we have identified that we have carbapenem resistant mechanism is there in the pathogenic bacteria, yeah, resistant bacteria. So what is the solution for it? So we are working in my lab with different aspect of it, like we are targeting some resistant determinant, we are targeting the host pathogen interaction, we are identifying some novel target for this targeting this Acinetobacter baumannii, we are targeting biofilm persistent. But in this talk I'm briefly explaining the three approach, how we can target the resistant determinant, how we can use this nano herbal approach to control this pathogen, and how we can target source response and iron regulation. So the first strategy I will explain about the resistant determinant. So if you see that I already told you that there is an efflux pump, so that is involved in the efflux of the antibiotic. So we have basically designed some drug for this efflux pump, inhibitor for this efflux. So what we have done, we have taken the protein of this efflux pump, that is ABC complex, and then we have selected different protein from FDA library, secondary metabolites, and then JIC library. We have prepared it and perform admit analysis, and then virtual screening, and then finally we have selected the lead compound by MDS analysis. So what we have found that if you see here, as I already told you that we have this meropenem or carbapenem, any antibiotic. They are meropenem and carbapenem, both are this carbapenem group. So if you give this carbapenem antibiotic, they accumulate inside the bacterium. But because of the presence of these efflux pumps, so that is basically pull out of the bacterium. So this bacterium become resistant to this. So but what we have done, when we have designed this mutant basically blocks this efflux pump, and then the concentration of this meropenem, which is effluxing this antibiotic, so which is ineffective when we have the efflux pump, is going to be very effective. So this is one thing we have done. And if we have we have compared it with the mutant of this ADB, and we have found that the mutant protein is going to be having same effect as our anti this this inhibitor of it. It clearly shows that we have what inhibitor we have designed. So this is not a published data, so I'm not explaining the detail of it. It's just brief of it. So it's going to be very effective in controlling this MDR. So because we now know that this EPIs work in the mutant and in this bacteria, wild type bacterium. So we wanted to basically whenever we are giving antibiotic, it develops some resistance. So we wanted to have a multiple mechanism of this efflux pump. So we have capped this this EPI with a silver nanoparticle, and silver nanoparticle is known to basically have the ROS generation and other things. So what we have done, we have capped it and then transfected, then use it, and then in the experiment we have used this capped nanoparticle, and we have found that, as we already proved it with the mutant and other analysis, so that this is going to block this efflux pump. But this, because of this silver nanoparticle, we are producing the ROS also. And one more thing, what is very interesting, what we have found, this EPI also changes the membrane potential. So if you see that, I have not explained in the introduction, but these efflux pump are requiring this proton to be functional. So this EPI is basically blocking this efflux pump, changing the membrane potential. At the same time, they are also producing ROS, so that this capped nanoparticle is very effective in terms of controlling this efflux pump based antibiotic resistance. So this is one approach. And the second approach we have.
Professor Dr. Tiwari, I'm afraid we are running short of time.
So in
Can you
5 minutes,
please,
I will stop.
please?
Okay, sir. Okay. So in second approach, because I am making it very brief. So in second approach, as you know that whenever we are giving any antibiotic, there is a DNA damage. Because of the presence of the source response, which requires this RAC active protein, is going to be repaired. So when we have antibiotic, the RAC source response induce the RAC, so it will repair it. So antibiotics become ineffective. So we have designed an inhibitor so that if we give antibiotic along with this RAC inhibitor, so the source response is not induced. So what will happen? It's going to not revert the effect of antibiotics. So we can clearly see in that if you are giving this DNA, you can see that if you are giving imipenem or any H2O2, there is a positive control. So if you are giving imipenem alone, we can have some DNA left here in the bacteria, but when you are giving along with our leads, there is a clearance of this bacteria, which clearly shows that the combination of this RAC inhibitor along with the antibiotic can be a one additive. The as I already told, there is an overexpression of Phe receptor. So we have designed this inhibitor for the Phe receptor also, and we found it's a functionally active. And then in the another approach, I told you that what we have done, we have designed hybrid nanoparticles. So what we have done, we have taken a secondary metabolite from the active plant. We have designed a nanoparticle. We have created a hybrid of it, and then perform the analysis. What we have found, quickly, I will move to the what we have found. So we have found that the Phyllanthus emblica have a gallic acid, is a secondary metabolite. So if you cap with this hybrid nanoparticle, this PVP cap nanoparticle, and there is a hybrid that is having gallic acid along with the PVP, so it's going to have very good effect. And you can see in the this same analysis that if you treat this Acinetobacter with the hybrid nanoparticle, there is a complete lysis of it. So it shows that it may be one of the good approach to control this bacterium. And in the last approach, because it's very difficult to find a suitable disinfectant. This the point of disinfectant is come into the picture. If you see the COVID-19, we cannot use all the disinfectant. So it's very important to find a suitable disinfectant that can be used in the hospital, where we have the chances of the epidemiology of this Acinetobacter baumannii. And what we have found that if you use this chlorhexidine, 32% microgram per ml chlorhexidine on the liquid solution, is going to be effective, and the solid surfaces 2.5% H2O2 is very effective to control this Acinetobacter baumannii. So what is the conclusion? In proteomics, we have found there is an overexpression of different proteins like beta lactamases, metabolic enzyme, energy production and iron transporter, and protein folding proteins. And we have found down expression of OMPW and other transporter. We have clone expression OXA51, and we found it's functionally active. We have developed this EPI cap nanoparticle, which is very effective in inhibiting the efflux activity of ADB efflux pump. We have designed an inhibitor for a RAK, which enhances the antibacterial activity of other antimicrobial. We have developed gallate polyvinyl pyrrolidine cap hybrid nanoparticle to control this carbapenem resistant infection of Acinetobacter in Parmoli cell line. And we have also seen that chlorhexidine, the liquid solution, and H2O2 in solid surface may be used as a disinfectant to control this Acinetobacter. So all these what I am presenting, except the EPI result, is got published in some or other journal. So my research work has been recognized by the Indian Academy of Sciences, different Indian Academy of Sciences. So I received INSA Young Scientist Award from this one, then associateship from Indian Academy of Science, membership from NASI, and other award from ICMR. And the research support is done by the SCRB and UGC and CSIR. So I would like to acknowledge Dr. Rajeshwari from AIMS to allow me to use this CD and fluorescence for biophysical studies, Dr. Arti Kapil to provide the clinical strain, Dr. Naidu Subbarao for helping me in bioinformatics work, and one of my collaborators from Italy who's helping in the biofilm persistent analysis, and my PhD student Privita Deepika Mona Lisa Vannayan Sarosh. Thank you very much. So this is all about this Acinetobacter baumannii.
Thank you, Dr. Tiwari. I think we are running behind the time. We are already late. There is only one question which has come up, and this is from one of our esteemed faculty, Dr. Kapil Lawrence. He has asked question: Dr. Tiwari has observed distinct changes in membrane morphology of the resistant microorganism. Can he throw some light on the variations or changes in the phospholipids of the bacterial membrane in the
Yes, yes.
resistant strain?
So that is very good question. So now we have started the lipidomics of the resistant strain and this one. So because that will give us a more clear picture. So we have started the lipidomics of the resistant strain compared to the sensitive strain, as we have done in the proteomics. So that might give us some more information about the phospholipid chain. But that is a very good suggestion.
Thank you, Dr. Tiwari. It was very informative, but you have covered a lot of area. I'm really surprised in this. You finished all the work in and
So I just
presented in
try to
very,
brief it.
yeah, very nice presentation. Next, auth- uh, next scientist who is going to present his work is Dr. Haim Chandra Jha. Dr. Haim Chandra Jha is currently working as assistant professor at the Department of Biosciences and Biochemical Engineering, IIT Indore, India. He has completed his PhD in 2010 from Institute of Pathology in Delhi under the supervision of Dr. Aruna Mittal, where he has worked on molecular diagnosis and pathogenesis of chlamydia pneumoniae in coronary artery disease patients and awarded PhD from BITS Pilani, Rajasthan. Then he moved to University of Pennsylvania, US for postdoctoral research, where he joined Professor Earle S. Robertson group working on tumor virus, specifically Epstein-Barr virus and Kaposi sarcoma virus, Kaposi sarcoma associated herpes virus. He joined IIT Indore in July 2016 as Ramanujam Fellow and joined the post of assistant professor in February 2017 and head of the department in August 2018. His current research work focuses on tackling important biological questions like how Epstein-Barr virus co-infection with Helicobacter pylori leads to the aggressive gastric carcinoma and drug resistance. What are the biological mechanisms behind this co-infected patient's higher mortality compared to other gastric cancer groups? Also, he has been looking for the role of Epstein-Barr virus, which is involved in several neural diseases like multiple sclerosis and Alzheimer's disease. Till now, he has published more than 60 research papers in the esteemed journals. His research group is actively working on SARS-CoV-2 virus and exploring new avenues in this domain. So it's a lot of work he has done. 60 research papers tells that he has done a lot of work, and he's working on Epstein-Barr virus, as well as he started on novel coronavirus also. Dr. Jha, please.
Thank you, Professor Kant, and thank you all, Dr. Tripathi, for giving me this opportunity. And yeah, it's a little bit going a little bit delay, but I will try to wrap up. So what I want to say, what kind of work we have done last 6 to 8 months in this pandemic and on the SARS coronavirus 2. As most of us know, this is the SARS coronavirus 2, the severe acute respiratory syndrome coronavirus. And till 14 December, almost all countries, like 218 countries affected. Total tests like 9.4 billion, and India also 1.5 billion, and confirmed cases 72 million worldwide and more than 9 million in India. And death is more than 1 million, and in India more than 1 lakh now. So our slides are visible?
Yes, sir. The slides are very clear. Your voice is also very clear.
Yeah. So symptoms and transmission of COVID-19, although almost all of us know, but so I don't want to go in this slide. Here a little bit I want to talk about the some factors, those that possibly associated with the SARS-CoV-2. It could be like temperature, although only few studies has been done yet in vitro, but some more study needed. So where is showing that a favorable temperature is for the SARS-CoV-2, either it is low or it is high, may alter the spread. Similarly, humidity, very high humidity may suppress the SARS-CoV-2 transmission. And then some other direct mode of the infection as well as the indirect mode. And if you talk about the biological level, so in the right side, this panel, in the biological level, there are almost 4 receptors. And very well known, we know that AS2, like everyone now, we know, okay, AS2 is one receptor for SARS-CoV-2. So along with that, TIMP, RSS2, neutropilin 1, and integrin. These four are also all three are also important for the entry of the SARS-CoV-2. So if we talk about here entry part, cell membrane, so there are all these four structures, small tiny structures have been made. So this SARS-CoV-2 can use any of these four, and depends on some cells are even positive for all four, some are even for two, high and low, but then most all the endogenous origin of organelles, they are having positivity for one of these receptor. That's why we know that multiple organ failure or most of these vital organs, either the heart, lung, kidney, or many other neural, everywhere this SARS-CoV-2 is able to infect and transmit very fast, because they have their receptors in all the cells. So basically this virus is coming inside, and then this is the RNA viruses. It start RNA virus replication through the translation, and ultimately it make the, with the help of non-structured and structured protein and Golgi protein, and it egress and make the new viruses. So almost all cell machinery it can be used, and it can this virus can survive. So this is the actual gist of the this picture. Now, if you talk about the molecular diagnostic tests, these are really very important, because initially in initial few months, even we don't know how we are going to go for the test. But then yes, real-time PCR it came out, and then we are almost taking throat swab and doing the viral genetic material, and these are the very high specificity as well as sensitivity also. Also ddPCR, digital droplet PCR, is also in the some research paper, and they are watching for this because of the more specificity and sensitivity, but it is very costly also at this time point. Then chest CT. We know that initially when there is no real-time PCR, things get standardized, because it took sometimes several months in India as well as other countries also. So initially doctors, they have seen chest CT only, and by the lung morphology and the pathophysiological changes, they start doing the identification. So till date in India, all kits are weighted by the ICMR for the real-time PCR. Total 6 ELISA and 15 rapid antibody test kits has been approved by ICMR, and ELISA and rapid antibody tests are not recommended for diagnosis till now. So antibody based test not recommended. Yes, antigen based test recommended. Why I was previous. So why I included these slides here, because this is very important. What kind of screening we are doing, and what kind of kits we are using, that is really very important, because all these instrument wise kits as well as the techniques, they may give us various results. As we just heard last, I guess 2 weeks ago, that some of the states in India are doing a RAT test, that is a rapid antigen test, and many of them, they are doing the real-time PCR test, and the sensitivity of the RAT test is 2.5-fold less than the real-time PCR test. That is in India. So maybe you are positive, but then if you go for the rapid antigen test, you may not be the positive if the viral load is not high. So these things are also important. Even we know that many a time some of the people are positive in one place and not positive in the same day on the another place, because the kind of kit they are using, the kind of primer they are using, they may also differ. And then that's why this is very critical. So for diagnosis, the proper pre-analytical issue are like proper collection of sample. We have to take nasal swab. So if someone, many of us have the problem in the nasal part also, so that if it is coming with the blood, then it is highly possible that some other contamination kind of stuff is going on, and we are not able to give the right result in first time. So the proper collection of sample, monitoring of patients with severe COVID-19 pneumonia, and safety during sample collection and preparation. Analytical issues are selection of assay. This is very critical, and because we have seen that some of the kits that ICMR has to refuse, even initially they pass out, because the positivity or negative are in the question. Because we know that this is the virus, and we are here scanning the viral genes, and all the viral genes cannot express all the time. Some viral genes may express early, some viral genes express may in the late, or some all the time. So that's, we have to be understood that what kind of gene we are taking out. then selection of proper molecular detection technique, and then target of QRT PCR, and the post analytical or interpretation of result and confirmation of infection. This is also important interpretation of result, because as I was in the initial few months in the here one of the helping one of the hospital, and I was surprised, because if out of 40 cycle, 35 cycle they say the cutoff for the positive. So if someone is 34.2 is positive and 36.4 is negative, you think that in one well, if you do real-time PCR, again you will get the same result? No. So we have to think about this, and then that's why some people are positive today and after 4 days negative, or some people are negative today and after 2 days is positive. So this kind of logistic has to be there, otherwise a lot of chaos, and we are not able to control it, and we are not giving even the right information. We are in the borderline. These are just some glimpse that we have involved in the screening process initial days. So standardization, safety, data analysis, all these are really very critical for the SARS-CoV-2 and the non-conventional approach. I'm just going because the time is a little bit short, so I'm going on our work only from here. So now the COVID-19, because they were locked down, everyone in our home, all students are in the home. So we started thinking that what kind of stuff we can contribute in this pandemic. So we started with some statistical data. Those are published initially in the Europe and some other country, or Europe, UK, and USA. And then we started also doing in silico analysis. Although my field was not in silico, we are truly virology lab and infection biology lab. But then we started making collaboration with the IIT Indore and other places also, and we start thinking what kind of understanding we can have for the SARS-CoV-2, either in the mechanical as well as for the therapeutic, and then some other part also. So I'm going to. So here just some glimpse about the statistical analysis. So we did like from starting of the infection, all these 10 countries. From the March 22 to till March 22, like from the day one infection, like in India, the first case was January. So similarly, some other country have like even the first week of January, some of the second week of January, someone having the first week of Feb till to 22nd March, because that period is very critical and everyone is like inside home, and we all are very scared. And then second phase, March 22 to March April 20, complete lockdown in India, like zero tolerance in the terms of lockdown. So what kind of problems happen? So what we have found that in the initial period of COVID-19, spread most of the countries except South Korea conducted very small number of tests, like in India also, because we don't have that much facilities and that much infrastructure. So it took time to develop all these stuff. But only South Korea is a very small country, or they have well-equipped, well-versatile health setup. So they have done very well. So low rate of screening might have contributed to significant increase in COVID-19 cases. That because you are not able to trace, you are not able to contact tracing, and that could be the possible to initial exponentially increase in various European countries and many other. So number of COVID-19 tests has increased significantly in India, but the test per million population is still lowest. This was data from till April 20th. So then we also compared the management, how they did, like death as well as the infection status. So what we have seen, the number of COVID-19 cases per million population has increased significantly in USA, Germany, and UK in the second half, like till April 20. Increased the COVID-19 cases in the South Korean Meanwhile. So basically those countries has included more tests. They are able to better control in initial phase also, but some of the countries they are not able to control in the initial phase. Then they have done a lot more tests in the second phase, like till April, or you can say till May now, and then they are able to control very well June onwards. So testing is really important. But then we are talking about the these countries like in terms of diagnosis and lockdown. So if you can see, even all the countries don't have very rigorous lockdown. Even some countries, even they don't follow, but still they control this spread. So diagnosis is definitely is important. Lockdown is also like it can slow the progress of the virus initially, and it help you like government. It help governments to prepare all infrastructure to tackle it. Either it is a hospital or the COVID hospital or the diagnosis center, all these stuff, or the treatment regime for the doctors. So they got time, a lot of time. That's why we can say the mortality rate in India is very less compared to the many very well developed country, because although our health system is not comparable than the UK, USA, and the European country, but still we did very good compared to others. So that lockdown helped us to prepare and understand how we are going to deal it. Now this is the second data, because initially we all know that, okay, this is the old age virus, everyone is in the old age, they are having problem, and then male also, some males are more prominent for that. So we have done this analysis, and it's already accepted but not get online yet. So what we have seen, no, this is not the male virus kind of stuff, like that kind of phenomena. Yeah, definitely the reproductive age of the female, they may have some positive side, like enzymes, like the hormones, they may protect them. But if you talk about the old age women, they are not get protected, and either the mid age, they may have some advantage. Like in Australia, if we talk about the confirmed cases here, so sorry, this is the first one that we are talking the age wise, like 0 to 19 year, 20 to 49 year, and 50 years and above. So we can see here that it is like 50-50, like in Australia and Germany, like approximately 50% are in the before below 49 year. Germany also. In India, yes, I think the population distribution is also very high in this age group, like 20 to 49 years. That's why more exposure in this group. If you talk about the Italy and Netherlands, yes, because these Italy is like initial country, those get affected. So here like 72% and 74%, because their demography, because most of these people in these areas are the old ages, like more than 50 years. So that could be one of the possibilities. Yeah. So basically, this data suggesting till I guess July that yes, the exposure or the infection pattern is not age specific. And second thing, it's not the gender specific also. So that for the gender specific, yes, we have here male and female, and we can see here in India, if we talk about India, Australia, India, Norway, and South Korea, all are having different setup. In India, see very contrastly, 20 to 49 year, the female in this age group is very less in the 20 to 49 year, but in the 50 year and above, females are very high chances of getting infection till July 2020. But if you talk about the Norway, things are like ups and downs. So there is not a fixed pattern. It depends on the society, how their people are, well equipped and making hygienic maintenance and understanding their education and their personal immunity. These things are only matter. It's not matter that what gender and what age you are. Now, if we talk about the biology, the SARS-CoV-2 led multiple organ failure, as we have seen a few slides ago, that they are having 4 major receptors, and possibly these receptors are found in all these organs like lungs, heart, brain, eyes, nose, liver, kidney, intestine. That's why possibly this virus may enter or accelerate the problems or pathogenesis in any of these organs. So our initial data where we collected from the hospitals, European countries, and there we have seen the comorbidities like kidney disease, liver, immunodeficiency, neurological, as well as the heart and lung and many others. So what we have found that significant population of the disease having the kidney disorders. The possible higher association of some disease like might be due to the altered physiological conditions of an individual or possibility due to the side effects of the drug. So it is not only the person, because what we have seen, the drug initially that is given, 6 to 8 drug, almost more than half are having major or severe side effect for the heart disease as well as with the blood pressure or diabetes. So when person is going with the COVID-19, mostly in initial phase out, people are, or doctors or clinicians, they are giving treatment for the COVID, not for their own comorbidity. So if someone having heart problem, we need to take care of the heart as well as the COVID. It cannot be, so many of the people, those are having died in this paper we have seen, I'm not showing the data here because of the slide limitation, but we have seen that even 75% death in total, like 100% death up to 75% in some of the hospital in the European countries are only because of the heart attack. So if some person having already some complication in the heart and then you are using the drugs, those are prescribed initially, they are simply fallout for as a death. So that's why the possibly initial too much, death because of the COVID, because of the can say the medication. And we don't know that how we are going to control it. But in India, we had done very good in that sense. Now, if we talk about not the clinical part, or the mutations, as we know that the one mutation that we are getting information in the everywhere in the newspaper and many places, the D614G. So this mutation is really very hit, and we have also collected data, I guess, till July from almost all the countries, and then we have found that more than 70%, those are reported like a sequence, they are having this mutation, including India. So this mutation is really, really very important. Although we have not done any in vitro experiment with this mutation, but some of the people, when they collecting data in the foreign countries, they have said that this mutation possibly one making the more viral load, higher viral load, as well as transmission more easy. So basically it help in the adaptation of the virus in different geographical regions, possibly. But we have not done that kind of experiment yet. And so there are some other mutations also we have found out, but this mutation is really very hit. And this is the data, I think, that we have shown from some other paper. And then there with the S protein, N protein, RDRP, PLR, and the ORF3A, they are the main SARS-CoV-2 protein, and the kind of mutation. So this is, we can see here, D614G, almost all Chile, Egypt, Germany, India, USA, almost everywhere, this kind of mutation is majorly reported, and some others. Now, this mutation in various protein. So see, like if we talk about, some of the A, B, C, D. So A is like E protein, B is M protein, C is S protein, and D is the RBD region of the S protein. So see, in the C, almost this mutation is, we can say, more than approximately 80%. So this S protein, that's why almost all people, those are getting infected, almost 80% having this, 84% is D164 having the D164 mutations. Now, what could be the changes? Why this is important? Till in silico analysis, what people found, and we have also understood that this D614, that is wild type, if changes with the G614, residue of chain A and the chain B, residue in the S protein. So the D614 of chain A with the up RBD conformation interact with the T6869 and K854 of adjacent chain, while G614, the mutant one, do not interact with any nearby. So basically, if just one this mutation changes the interaction pattern of S protein, and we know that the initial transmission of the virus, this S protein, S spike protein is really important, has been known. And that's why, like, we are washing our hand with the soap and many other, because this S protein have the lipid bilayer outside. And if we are basically, we are thinking that if we washing hand with the soap or some kind of sanitizer, so we are destroying that lipid coat, and we are destroying the S protein. If we are destroying the S protein, basically the transmission of the virus is delayed, or basically there is no occur. So,
Dr. Jha, I'm afraid we are running short of time. Can you please?
Okay, sir. I'm just
wind up?
Yes, winding in the next 2-3 minutes. So this is one of the comprehensive picture that just appeared in the Helion. So there is 4 different mechanisms how virus can enter. Like first one, clarithrine mediated endocytosis. So here ACE2 and all these late endosomes and viral replication occur, and again it binds with the new virus. Second one, the clarithrine raft mediated endocytosis. Third one could be the integrin mediated virus, that is very well known. And fourth one, myristoylation. Basically myristoylation in the protein, that may also help. So basically this is the comparative picture, and if someone wants, they can check this manuscript, structure and mutation analysis, SARS-CoV-2 in the Helion. And then treatment and vaccine, as is also important. So many of the people, they are talking about the novel drug discovery, and as we know that this is in short time period making the vaccine, and there could be lots of problems. So drug repurposing could be one of the possible ways. In that way also, we have seen that some of the drug, how they are acting, like some of the drug important in the entry, some of the drug are important in the translation and the replication. So in that way, how we can contribute. So we took the Himalayan plants, and then we did search against all these body organelles, their property, as well as the SARS-CoV-2 proteins. And we have found some of these plant compounds, and some in silico study that with the help of other people we're doing. I'm not going into the detail right now. And we have also reviewed some of the gastrointestinal, because many of us know that when the SARS-CoV-2 infected, not only the oropharyngeal, the gastrointestinal system also get problematic. So what are the consequences? What could be the markers, possible markers and receptors are that we have included here in the gastrointestinal? And then second thing, if virus, as we know, that can be enter the neural system, because from the gut, not only one part, it could be going by the brain directly, or by the olfactory sensory, or by the mucosal, or by the enterocytes, by the our gut. So all these four ways also this virus or their response may enter into the neural part. And after when we go into the neural part, there is huge cytokine storm, and that cytokine storm may lead to various deleterious effect in the future course of action. So that kind of stuff we have also included. So as the time getting short, I would like to acknowledge my research team here, and then my funding agency, IT Indore, CSIR, DST, MHRD, and Instrument Center. So this all these works are included from like here. The left side, right side is me, and then Anu and Dharmendra, Omkar, Diksha, Shweta, Nidhi, Tarun, and Charu. My collaborators, my US collaborators, and Indian collaborators. And yes, Dr. Tripathi, we are also closely now start working with the SARS-CoV-2. Thank you.
Thank you, Dr. Jha. Your lecture was very clear. I'm surprised in such a short period you have done a lot of work on SARS-CoV-2. Just all people were busy with the lockdown and you were working. Really admire your tenacity in doing all this work during this lockdown period, and you have done a lot of work. There is no question from the audience side, but I have a question. Recently I have seen 2-3 disturbing news. One was that SARS-CoV, particularly, it was affecting cardiac myocytes. That was very alarming situation. And another one, just recently, in just 2 days back, there was news that it is affecting the eye. The co-infection of fungus after SARS-CoV infection is. What are your views on those?
Yeah, as we have shown one of the slide where these receptors, what we are talking about, the 4 receptors, they are present in almost all the organelle. So eye and many other organ, it is possible. And yeah, infection of from the eye, that's why we are taking the seed. So that is known, like it is possible.
Thank you, Dr. Jha. It was very nice presentation. I think people enjoyed your presentation a lot. Next, we have lined up Dr. Professor Sarvas More. He's going to talk next now. Professor Sarvas More is currently working as the head of the Laboratory of Immunogenetics, VU University Medical Center and at Amsterdam, and as a director of the Institute of Public Health Genomics, University of Maastricht in the Netherlands. He has been working for at least 25 years with a central focus on female infertility and women's reproductive health. He is an authority in this field and has published more than 200 internationally reputed research papers, 200. He has also served as the director of scientific consortium of the European Framework Program 6 grant on functional genomics to understand chlamydia trachomatis disease with 20 European, African, and US groups. He is visiting professor at the Department of Molecular and Cellular Engineering, Jib Schwartz, that's our university. He was conferred PhD degree by the VU University, the Netherlands, in the area of epidemiology, transmission, natural course, and diagnostics and immunopathogenesis of human urogenital chlamydia trachomatis infection. He is a serial entrepreneur in the field of infectious disease, diagnostics, human biomarkers, and personalized medicines to generate new application in healthcare system. He is doing a lot of work, Dr. More. Now floor is, I'm handing over to Professor More. Welcome, Professor More.
Professor Kant, Thank you very much for the introduction. Can everyone hear me and see my slides?
Yeah, we can see your slides as well as we can hear you clearly. Thank you.
Excellent. I would really like to thank the organizers very much for providing me the opportunity to give a seminar on one of my favorite topics, which is infertility research. And I will tell you something about how IVF failure can be predicted using the vaginal microbiome. Currently, like Dr. Kant was telling, I'm working at the Laboratory of Immunogenetics in Amsterdam, which is a lab mainly focused on biomarker discoveries. I work in the Institute of Public Health Genomics, which is an institute looking at how to translate genetic and genomic findings into healthcare systems. And finally, there's a series of spin-off companies in which we translate those findings into diagnostic applications. I think one of the main focuses of our companies is women's reproductive health lines. The first one focuses on sexually transmitted diseases. So we have developed assays, which we deliver to laboratories and hospitals. We work on direct-to-consumer testing, meaning you can order these tests as a person online. Once you know that you have had a sexually transmitted disease, there's a high chance of subfertility or infertility. This is for a major part based on your genetic composition, how you battle infections, and how you deal with inflammatory processes which could result in tubal pathology. In level 3, you have actually a stage where your tubes are damaged and you cannot get pregnant by yourself. So we do vaginal microbiome profiling to investigate something about the chances to become pregnant normally. And finally, we work on adapting the vaginal microbiome, and we also work on recurrent miscarriage using microbiome research. And today I will tell you something about level 3A. So implantation failure prediction. Microbiomes are normally assessed by next generation sequencing. However, one of our companies has made a completely new technique focusing at the IS, the interspacer fragment between 16 and 23S. The interesting thing is that a lot of microorganisms have multiple repeats of the 16S, 23S internal spacer part, but they are not equally in length for the internal spacer, meaning one microorganism will have multiple bendings. If you look here for an example, there are even microorganisms who have up to 5 bands. These bands are completely unique for each microorganism. To further distinguish all the microorganisms available, we do a PCR amplifying the IS fragment with 3 different colors, separating 3 different sets of specific bacteria. This gives us a direct overview before we go into detail of each bacteria within those 3 clades. What you get is a bending pattern, which on the x-axis says something about the length of the fragment. The more to the right, the longer the IS fragment will be. On the y-axis, you see the quantity, so the amount of specific microorganisms in here. You also see in this bending pattern the 3 different colors for each group. The whole process from getting a sample, isolating the DNA, making the IS profiling and do the analysis, it is done automatically within 5 hours. If you want to read more about the technology, you can go to one of our websites on InBiome or IS Diagnostics. To validate this technique, we looked at 300 women with fertility problems who would enter an IVF procedure. The 8 large hospitals in the Netherlands were all participating in this study, and the Erasmus MC was the coordinating study group. We had very well-defined inclusion criteria, very good defined exclusion criteria to have a homogeneous group of women. Since next generation sequencing is the golden standard as compared to our new patented ICE Pro technology. We compared our technique to next generation sequencing before enrolling the whole study. You can test two types of clinical materials, urine and the vaginal swab. So we had a comparison of two techniques and two different swabs. Without going into too much detail, the blue blocks on the bottom is V for vaginal, U for urine, and you very quickly see that the amount of microorganisms in a vaginal swab are way higher as compared to urine, though the composition is quite identical. So based on this, we have chosen to do the vaginal swab, which is also more logical because it's anatomically more closer to the uterus. This is a very complicated picture. I will not go into too much detail, but you can see the three colors of the three different primer probe groups. And what you can immediately see on the right is that it's quite different as compared to the two first blocks. The first block is Lactobacillus iners dominant, the second one Lactobacillus crispatus, and the third one has almost no Lactobacillus dominant profile, and the bacteroids are quite high, which is the red dots. When you look at those profiles a little bit more in detail, what you can very easily see is that the non-pregnant women have a different microbial composition, both in species and in amounts, which means that based only on the vaginal microbiome, you can make an algorithm for prediction of failure of IVF. And you can also look at the composition on which compositions have the highest success rate. The end result is that there is a score, which we call a receptivity score, with a very low clinical chance, 5.9%, which means that 1 out of 20 women will fail. Yes, in the medium, around 5 out of 20, which is the normal score of 25%, will be successfully. And then there is a high score with pregnancy rates up to 52.6%, while on the normal conditions in most countries, pregnancy rates are around 23% by the first cycle. In the end, we will provide an overview to the clinician currently with in which profile the women fit. We give a profile with a score, in this case a low score, less than 6%, and we give a more detailed score on which microorganisms are present. This is all coordinated by a company called ArtPret. So currently, the receptivity assay is being used. These are the boxes which are at the different gynecology centers. And around 25% of the women is currently giving advice not to proceed with IVF with a clinical accuracy of 95%, since these women will almost have no chance for becoming pregnant. The remaining 25 women, there are different options. First of all, we are currently inside an investigation to look how stable the vaginal microbiome is. And looking at a follow-up of women with an abnormal microbiome, taking samples every month, we see that in around 2 to 3 months, almost 70% of the women change. If you wait longer and the aberrant vaginal microbiome is still present, they do not change. So we are now defining that around 2.5 to 3 months, the norm will be defined within the upcoming 3 months. We give an advice to wait, do a new microbiome profile, and then proceed to IVF. In the end, out of the 25%, 1/3, around 30%, will still not have a profile to become pregnant. This small group of around 8% of the total initial group, you have to do something else. And for that group, we have currently a patent portfolio and an approach how to change the vaginal microbiome such that it is more a normal microbiome with a normal lactobacillus dominant profile, in such a way that pregnancy is still possible. I think a lot of you have heard about microbiome transplants. The most important one and tested one are the fecal microbiome transplants with different microorganisms. There has been a universal donor screening approach for vaginal microbiome transplant. So in theory, it's possible, but it's not yet done as far as I know, but it is something to consider. There are some very good publications in Nature Medicine and Nature Reviews Microbiology on how that could be done. Currently, we do not work ourselves on this approach. We currently work on different approaches, microbiome adaptation via pre and probiotic approaches. The current state of our project is the following. The 8 large hospitals in our country participate. An insurance company is currently reimbursing 1,000 receptivity assays, which are 330 euros in our country, as compared to an IVF procedure, which costs 3,500 euros. For an insurance company decision, it's an easy one because you directly save over 90% of the costs for not providing an IVF procedure in those women who have a very low chance for becoming pregnant. What we are hoping is that the receptivity assay based on the current study, which we are evaluating together with our reimbursement company, that the same prediction rules comes out of the re-evaluation, that the test acceptability to women is as high as we claim, such that the reimbursement will take place in our country. And like I said, we are working on vaginal microbiome adaptation strategies, which are based on pre and probiotics with oral intake. We're also searching to expand this assay to other countries and other continents, and we're searching for new collaborations. Like in many of these assays where you look to either genetics or to specific partially genetically and environmental determined variables like the microbiome, it usually means if you expand this, for instance, to India, once you do the study in part again, which means that the prediction rule will potentially stay for over 90% identical, but that perhaps a little bit of other microorganisms or other compositions need to be added to the current algorithm. So what is now happening is that in 1978, the first IVF took place. It was around 10% chance for becoming pregnancy. When the ICSI came in 1991, it was 20%. When the IVF culture medium and embryo quality was becoming better in 2016, it was 28%. And currently, taking the implementation factor of the microbiome into account, we almost reach a 50% chance of becoming pregnant for the first IVF cycle. There's a large team of people working on it. It's a series of professors from different universities. On the right, you see Rivka Kudoder. She is currently the PhD student on the project. She will defend her thesis in the beginning of 2021. And on the left side, you see Dries Budding, who is the clinical microbiologist and is the one who has made the IS Pro technology possible. And if you want to know anything more about how we translate our female reproductive health line from bench to bedside, you can always send me an email. And like I said, we're searching for new collaborations. Thank you very much for your attention.
Thank you, Professor More. This platform is open for questions. I think audience will get really benefited by asking questions because it's directly from lab to the setting up the patient-based industry. So you can actually go from lab to business model, and I think Professor More is the right person to answer all those questions. If you have any question, please do ask. We have Professor More with us. I think people will be typing their questions. I have a question, Professor More.
Thank you.
You probiotic that you are uh, having with you, is it region specific, like geographical region specific, or is it race specific, like mainly Caucasian, and or you have seen in other races also?
It's a very, very good question. When we look at the vaginal microbiome, we see that those women failing are actually consisting of three subgroups, more or less, which means that the composition which we are making, we would like to have three different compositions, such that it is a little bit personalized medicine approach. You are completely right that other ethnical groups might have small differences inside their vaginal microbiome. There have been done studies in the past which show that Chinese, European people, African people, they all have a little bit differences in their vaginal microbiome. So that completely means if we would like to translate our current algorithm and the current microbiome adaptation strategy, you have to do a new study in a country and see how much it actually differs and which adaptation strategy composition-wise would actually be the best. I completely agree with you. It is ethnically based, it is genetically based, and it is also in part, I think, cultural and nutritional based. So very complex.
Yeah, thank you, Professor More. It was a very nice lecture. I think a lot of people will get benefited. I wonder, people have this, might be thinking, particularly who are not from India. I mean, India, they are also wondering that we have such a huge population in India, and why someone should work on fertility area. But you can see the satisfaction on the face of a mother who is trying to get pregnant for a long time, and then she successfully gives birth to a child. Nothing, you cannot pay for it.
No,
Am I correct?
Yeah, you're completely correct. It's very weird. You will, in a few years, be the largest country, people-wise, in the world. You will beat China. So you would think there is no fertility problem. However, the fertility problem in India is actually in the top 3 in the world, even though you have so many people. And I personally feel like you were also saying that be able to become pregnant yourself as a woman, as a couple, it is very satisfying. So even though there are a lot of people inside India, I still think it's a very relevant approach. I have to be very careful saying this. I think in some of the cultures, not being able to become pregnant, it's partially stigmatized. And if you can change that in a way where you only have to take literally a pill with different compositions of microorganisms, including lactobacilli, in such a way that you can become pregnant, I think it's completely priceless. And that's the research which we are doing inside our university, inside the collaboration with you and Jonathan Lal at SUATS on female reproductive health. It's an extremely rewarding research line because you're really trying to change pregnancy rates among those who are unable to get pregnant. And it's a very rewarding type of research.
Thank you, Professor More. It was really very nice talk. Now we move on. Next speaker is Dr. Anita Krishnan. Dr. Krishnan, thank you, Professor More. Dr. Krishnan received her master's and PhD in physiology and molecular biology, plant molecular biology from GP Pant University, India, and bachelor's with honors in microbiology from Delhi University, India, She was a postdoctoral fellow at University of Minnesota Medical Center and Boston College before joining Harvard. Dr. Anita Krishnan is currently working as Director of Research and Development, Aviseda Therapeutics, Cambridge, Massachusetts, USA. Previously, she was a staff scientist at Harvard Medical School, Department of Ophthalmology for 7 years, where she managed multiple projects in different ocular diseases. She has worked extensively on neuroprotection of retinal ganglion cells using gene therapy. She has significant experience in working with a range of modalities, including gene therapy, small molecules, and antibody approach to target disease. Uh, Dr. Krishnan, she's going from a small molecule all the way to the gene therapy. Wow, it is something great in the area of biotechnology. Dr. Krishnan, please, floor is yours now.
Thank you so much. Let me just share my screen. I would like to thank the organizers for giving me this opportunity to present, and especially my senior during my PhD and master's days, Dr. Prakriti Mishra, who I was very closely working during my PhD days. So I really appreciate her giving me this opportunity to present some of my recent work. Like Dr. Kant mentioned, I am currently working in a company where we are trying to take things more to the clinic rather than just into the more in the research side. But today I'm going to be talking about some of my recent work on gene therapy for ocular disease, where we just published a paper in Nature talking about reversing aging in using gene therapy. So I started my postdoc at University of Minnesota looking into neurodegenerative disease of the CNS, mainly HIV/AIDS. So the main concept where we are working was in a drug abuse model. As we all know, that drug abuse is a significant issue throughout the world, and people becoming infectious due to the drug abuse, like the opportunistic infection, is very high. So what we saw here that in a combination of drug abuse and HIV, there is a significant infiltration of the bacteria into the blood-brain barrier, thereby activating the innate immune system, mainly looking into microglia, macrophage in the brain. And we saw that all these inflammatory mediators, which are upregulated, further leads to neurotoxicity and eventually leading to dementia or HIV-associated neurocognitive disorders. So this was something we elucidated back in 2007. With this concept, I then moved to Boston College looking into the similar concept in a non-human primate model. So initially we saw in mouse models, and then I moved to non-human primate models looking into the role of monocytes and macrophages in that. So then with that experience, when I joined Skepin's Eye, which is a part of Harvard ophthalmology department, I moved into looking into neuroprotection of the eye. So glaucoma is one of the key disease of the eye. So I started working on that, looking into the role of inflammation and neuroprotecting the neurons of the eye for vision. So as we know that eye is the most ignored organ. People don't pay attention to the eye unless you lose kind of half of the vision, and people just don't go to proper eye checkup and then take care of the eye. So we only go to the doctor only when we start losing vision. And that is very late stage of the disease. So sometimes if it's good to catch things earlier. And as we all know that the highlighted ganglion cell here is the neurons of the eye. And we all know that neurons, once they are degenerated, they cannot regenerate back. So that is why when we lose vision, it's very hard to get back the complete vision. So people are trying to see if we can restore that. And one approach was our approach, where we use gene therapy to see if we can restore the vision. And the eye has multiple layers of cell, and the top layer is the neuron, and then These are the photoreceptor layer, which are mainly the ones which help in transmitting the vision to the eye. So, so what is glaucoma? Glaucoma is a chronic neurodegenerative of the disease of the eye causing irreversible blindness. You start losing vision in the periphery and also in the center, and eventually you lose total vision. And it's an ocular disorder which is characterized by the progressive loss of the retinal ganglion cell and their axons. So the axons are the optic nerves which connect the eye to the brain. And once they are degenerated, you cannot regenerate them back. And all of the different forms of glaucoma have one endpoint, the ganglion cell loss, and that is due to apoptosis. As we all know, apoptosis plays a key role. In my previous lab, we looked into multiple pathways which can be targeted for addressing apoptosis. But the main issue in glaucoma is because of the multifactorial etiology, that it's just not one factor, it's just not inflammation, it's oxidative stress, it's protein misfolding, there is glial modulation, and excitotoxicity. All of these factors contribute to a disease. So it's very challenging to develop neuroprotective therapies for glaucoma or for any other disease for that matter. So then we looked into the clinical trials and other database to see what is done in the literature to see how people have targeted the eye, and we saw that there were no clinical trials or no gene therapy approach which was very effective in glaucoma. So when we started using this as one of our key approach. So how do you work on gene therapy? So what is the major approach? So gene therapy to treat glaucoma is to approach to treat or prevent the disease by augmenting, either you increase, in our case we did that, to replace or suppress one or more mutated genes with functional copies. That's the key concept of gene therapy. So this is where it shows that when you have a normal gene, then you have a virus. So in most gene therapy cases, we use something called as the adeno-associated virus. We use that, and then we augment, means like you increase, sometimes you can replace or even suppress the mutated genes, and then you have a normal functional copies of that gene for therapy. So in our goal was to do one single administration and then have a lasting therapeutic effect, because it's the eye, we cannot go and do multiple injections. So we have to make sure that we do one injection and we have a good lasting therapeutic effect. So because of the retina, you have to choose what AAV, that is the water adeno-associated virus, you have to choose, because all of them don't go and target the specific cells you need. So based on the literature, we chose the AAV2. It is the adeno-associated serotype 2. That is the one which was shown to achieve the best neural transduction. So when injected in the eye, you were able to see a good expression. It was able to provide long-term expression and then have a good safety profile. As we know that currently AAV is the only gene therapy approach which is FDA approved for the eye. So that is the most common way of targeting the eye using gene therapy. And so how do we do the injection? So it's the ocular injection. So it's not systemic where you can just do it in a muscle or in the vein. So what we do is we have two methods of injecting. We do the intravitreal. Intravitreal is going very close towards from the top of the eye. So that way you go directly into the cavity of the eye, and that way you're directly going and targeting the cells in the retina. And another way is going below the retina. So this is a more, not a very comfortable injection, because if the surgeon who is doing slightly misses your cavity, then it might cause major damage. But the intravitreal injection is the most accepted version of injection and most commonly preferred route for injection in the ocular delivery. So this paper here we published just came out early this year, where we were able to reprogram some of the youthful epigenetic information and restore vision. This was a big collaborative project with Department of Genetics, as well as David Sinclair. He is a person who works on aging, in looking into different other factors. But when we collaborated, we wanted to see if this was something we could use to restore vision. And so what our concept was, we used an OSK. So OSK is it's the Yamanaka factors, which is very commonly used for stem cell and other research. And we modified those factors and then injected them into the retina. And we saw that there was axon regrowth, as well as the ganglion cells were protected. So this was our basic concept, which we used for restoring the vision. And what we did was we used the OSK. It's one of those transcription factors from Yamanaka, OCT, SOX2, and KLF4. These factors were combined, and using gene therapy, AAV2 was genetically engineered. And then we did two different models for the eye. We did an optic nerve injury model and glaucoma model. And we saw that these factors, once injected in a 4-week-old mice, we saw that there was regeneration of the optic nerves. And that is something which nobody has shown, that there is no theory or literature which shows that you can regenerate nerves. So this was something which we showed recently, and it attracted a lot of attention because of something can be translated to the brain, because as we know that a lot of neurodegenerative diseases of the brain, like Alzheimer's and Parkinson's, the main issue is that you cannot regenerate the lost neurons. So this modification and this concept of Yamanaka factors, which can be injected, is known to regulate the DNA methylation in these cells and then changing their phenotype to a younger cell response. So that is how we kind of saw results. So, and now we are moving more translational, where we saw this effect in a mouse model. So now we would move into phase 1, phase 2, or other preclinical studies to validate these studies. So that is something ongoing over here. So, and with this, I would like to thank the whole group here. This is some of my key lab members, and this is people from David Sinclair's group, and also the Harvard Ophthalmology group for their support for this work. Thank you.
Thank you, Dr. Krishnan. I'm wondering, you have gone all the way from small molecule to gene therapy. I mean, you are completely covering the panorama. One question I have here, you have taken 4 weeks old mouse for your model, but generally they are too young. I mean, in there you are supposed to have these now, I mean, these cells are very young, so they reprogramming will be much easier. But in case of macular degeneration, the person is going to be a bit old. So what is your in that?
So this is just one figure. I mean, this paper had like so much work in it. What we have another data which supports that. We took a mice which was aged. So we took a 24-month-old mice, and what we did is we induced glaucoma into them. And then when we injected these vectors, we saw that the neurons kind of was protected. So it was protected in the way that you didn't like have from 10 neurons, we didn't go to 100 neurons, but the 10 neurons was able to restore vision, which was able to give the mice the sight, which was enough for him to see, the mice to see, because it's hard to regenerate the whole neurons which are lost. So we were able to protect the neurons which at a certain stage, that way there is no further degeneration of any of those neurons. So this figure here, you can see is an optic nerve injury model. So this is before the treatment, where there is, when you have the OSK, when there is no injection, you don't see any regeneration. So these are the regenerative neurons, the ones in bright yellow. So you see that in a mice, when you cut the optic nerves and they don't have regeneration, you don't have anything regenerating. But when you give our OSK, you start the regeneration process. This is where it goes to the brain, but you definitely start seeing some amount of regeneration. And we did RNA sequencing, and we found out the genes which were responsible, and we found that these genes were definitely the ones which are seen in a young mice. So that way we know that they are able to reprogram their dead cells to have expression of the young genes. So that was the whole concept here to see.
Very nice work. Very nice work. And very promising from patient point of view. Thank you, Dr. Krishnan.
Exactly. Thank you so much. Thank you.
It was a nice talk by Dr. Krishnan. Now we move on, and we have Dr. Susheel Mishra, who will be presenting his work. Dr. Mishra is currently working as a research scientist at the University of Mississippi and Oxford, USA. He is a skilled computational glycoscientist with a PhD in biomolecular chemistry and 7 years of post-PhD work experience in applying computational chemistry and bioinformatics to perform research in glycosciences. After finishing his PhD, he had successfully secured postdoctoral fellowship awards from the Japan Society for Promotion of Science, JSPS, in 2016, and Tokyo Biochemical Research Foundation Fellowship 2017 to research on modeling protein-glycan interaction through computation. He has worked with leading structural glycobiologist Professor Yoshiki Yamaguchi from Riken, Japan, and collaborated with Professor Naoyuki Taniguchi, also from Riken, Japan. During that time, he modeled Golgi, GPI, and galactosamine transferase structure in collaboration with Professor Taro Kinoshita from Osaka University, Japan. The structure of protein O-mannose, kinase, and lactin orisata, and co-authored several papers on similar topics. He was awarded the prestigious Marie Sklodowska-Curie Fellowship in 2017 for the project Smart Design of Recombinant Antibody Fragment Specific for Carbohydrate Molecules. He worked in the Advanced Glycoscience Research Cluster headed by Professor Lokesh Joshi on developing a workflow for enhancing binding affinity and specificity of SCFV fragments against certain glycoepitope. So Dr. Mishra is mainly working in the area of glycobiology. Unfortunately, a lot of biotechnologists, particularly molecular biologists, they don't realize the importance of glycobiology. And I think his lecture will enlighten us what is the importance of glycobiology. Particularly, he has worked on SCFVs, which becomes very important. Glycosylation is very important for immune function of these antibodies or antibody like molecules. Dr. Mishra, please, floor is yours.
Okay, can you hear me?
Yes, we can hear you. We can see your slides clearly. Thank you.
Thanks a lot. Thanks, Professor Rajiv Khan, for a kind introduction, and Dr. Vijay for giving me this opportunity to talk about my work. That's mainly about the computational glycosciences. So unfortunately, I won't be talking about those SCFEs because that work is still unpublished and we don't want to disclose some of the data. But I would be telling you about the computational methods that I had been using to model protein-glycan interactions and how effectively we could get into the minor details of molecular interactions using computers. So by now, there had been, at least after the COVID, we all know about that glycans play a key role in host pathogen recognition. And especially, you could see this is a structure of a lymphocyte. So our our thinking of the glycoproteins is that there's a protein and there could be a couple of glycans on this, attached to this protein, but that's not the reality. If you look at the structure of the lymphocyte here in the left side, the upper figure, you could see this black band of hair-like structures, and this is glycocalyx. So there is a really thick band of carbohydrates at the surface of the cells. And once these cells land into the host cell, the carbohydrate is the primary molecule that's going to go and attach, or that's going to be in the contact with the host cell. And they play a really key role in host pathogen recognition. And in these two structures, you could see these two bacteria, they have a really thick glycocalyx in the outer cell surface. And once they attach to the host cell, and these glycans play a key role, and the recognition occurs, and then they infect the host cell. In the right-hand side, you could see that these glycans, which are attached to the cell surface, they play a key role in different kind of molecular recognition. It could be cell-cell recognition, or cell bacterial recognition, or recognition of the antibodies with the cell, and different other things. And the proteins which recognize these carbohydrates, naturally, these proteins are called lactans. So I will be talking about the lactans a bit in my work later on. One of the most key important examples these days is the coronavirus. So this virus, this whole story started at the beginning of this 2022, at least. And in March, we got to know that this spike protein S here, this plays a key role in recognition. So this spike protein S is recognized by the human ACE2 receptor. But we didn't know that what's happening here until this structure of the spike protein S came out by a Chinese group sometimes later, like in late March. And soon after this paper, there was another paper showing that there are multiple glycosylation sites in this spike protein S. And a group of robots from Georgia, they mapped all those glycosylation sites, and they prepared a computational model that how the structure, how the glycans would look like in this structure. So you could see that the protein is almost covered by the glycans. So this is just a flexible, all possible conformation of that glycans in the site. So once this spike protein S comes into contact with the host cell, and it's bound to ACE2 receptor, you could see that the glycans, there's a high possibility that the glycans are the primary things which are going to be in the contact with the ACE2 receptor, and they play a role in recognition. And not only this, let's see, there are several papers, there are numerous reports, and it's well confirmed that the glycan can serve as a marker. So if you have a normal cell, if you have a cancer cell, you could differentiate between those two cells depending on the nature of the glycans they have. O glycosylation, so O glycans are often truncated in cancer cells. So you could see that they could not be elongated here. And N glycans are often core fucosylated. So this is the representation for fucose. They have a core fucose in their structure. And not only this, we all are aware that our blood group types are determined by the glycoproteins and the glycan present on it. So O blood group type has this antigen, A has this one, B has this one, AB has both of them. And that's how the blood transfusion is determined, because if we have A blood group type, we do not have a B antigen, and if we take a B or AB blood group type, it's allergic to us. Not only this, there's also this alpha-gal fragments that I was working on, and we designed SCF antibodies to recognize this alpha-gal fragment. And this alpha-gal fragment is absent in human, but it's present in many other mammals. And very often people who eat red meat, they get allergy. And that's because of this alpha-gal fragment coming to their body through meat. So that was kind of basic introduction that why I'm interested in glycosciences. But let's think what computations can do for drug designing, especially in the area of glycomics or glycosciences. We all know that the very primary step of drug designing is finding the hit and finding the lead compound, and these are finds by just looking at protein ligand interactions and which ligand can bind to the protein and how effectively and how tightly they can bind to it. So computations can be used to speed up the discovery of new hits. So instead of testing 2,000 molecules in the lab, you could just test 1 million molecule in computer in a very short period of time, and you could just pick some hit from the computational screening and just quickly test them in the laboratory. It can help us to optimize the lead compound. So once we get a lead compound, we often go for the second phase where we try to make the modification in this lead compound and we try to optimize the binding property. So it could help us in doing that as well. And then it could help us in reducing the off-target activity, so that we could try to see if this molecule binds to the other proteins as well, and we could reduce that. We have very selective molecule. My long-term research interest had been to apply, evaluate, and develop binding for energy calculation approaches that can help in understanding the protein glycan recognition, and they can help in protein engineering or engineering glycomimetics to create better recognition molecules. And I had been involved in developing free energy calculation workflows for protein glycan complexes. So if you look at the binding free energy calculation approaches, there are basically 3 kinds of approaches. First is the empirical scoring one, where you have a protein, you have a ligand, and you just don't care about the dynamics. You just try to see how a ligand binds to a protein. And then you have a semi-empirical approach where you run simulation of a protein, you run a simulation of a ligand, and then you try to see how both of them bind. There's a dynamics involved, and basically there's a linear interaction energy approach, or MMPBSA, MMGBSA, or QMMPBSA. And the third type is free energy perturbation, or thermodynamic integration, or potential of mean force based approaches, where you could compute relative free energy in the binding, and there are very rigorous computational approaches. They can take days or weeks to run in really strong computational power, like in clusters or in supercomputing centers. Today I'll be talking about a couple of my recent work, but if you would like to have some idea of how these empirical scoring or docking or linear interaction for energy calculation process work on protein glycan complexes, you could have a look in these papers, which I published back in 2012 or 13, something like this. But let's see how MMGBSA approach work on protein glycan complexes. So we all know that we are looking at the binding for energy. We have a protein, we have a ligand. Sorry, could you mute yourself? We have a protein, we have a ligand, and once they form a complex, there is a delta G bind. And this delta G bind is
Giderappa, that it is coming. Yeah, thank you.
Okay. And once we have this, we want to calculate delta G binding, we just compute the free energy of this complex minus free energy when it was in free form. They were in the free form. And that's how we get the free energy. But computationally, how we do is that we could run 3 molecular dynamic simulation, one of the receptor, one of the ligand, one of the protein, and we can collect the snapshots, their conformations, and then we can get their energy and calculate the difference here. But very often we use a single trajectory approach where we just run a simulation of a complex, and then we just separate the protein and ligand from this, and then we I applied this approach on lectin RSL, which binds to fucose So it's FICOs recognizing lectin, and it has 6 binding sites. So it's a trimer and has 6 binding sites. And I try to calculate the binding free energy of different sugars with this RSL, and I try to find that what settings or what workflow would work best for finding the binding free energy between sugars and this kind of lectins. So those who work in the computations, they must be aware that there are different settings in MGBSA calculation. So you could use different van der Waals radii sets. So here I compare those 4 van der Waals, 5 actually. There are 5, but we just 2 of them are very similar, so we didn't use them. So here I'm showing those 4 van der Waals radii sets. So if you use Bondi, M-body, or M-body 2 or 3, and this is the correlation between the calculated binding energy and the experimental binding energy. So we already knew the experimental binding free energy from ITC for those molecules. And you could see that the best correlation that we get is for M-body radii set. And the second bar is mean unsigned error. So the error between the calculated and the experimental binding free energy, so which is the lower. So we recommended that this is the van der Waals radii set that should be used to calculate the binding free energy of protein ligand complexes in general. There's also different generalized Born model that how solvent effect is handled in implicit solvent calculations. And these codes, they basically refer the author's name who published this generalized Born model. And you could see that the GBHCT model was actually the best among all, that this could give us the best correlation and at least minimum mean unsigned error. Not only do this, you could also run QMMM-GBSA calculation, where you could define a small Q quantum region inside the molecule. And you could say that, okay, this certain number of amino acid residues and the ligand will be handled by the quantum mechanics, and rest of the molecule will be handled by molecular mechanics. And you could get the binding free energy. Often it happens that molecular mechanics it's a very simplest model, simplest among all. So sometimes we have certain interactions that could be underestimated in molecular mechanics. So you could use quantum mechanics to estimate those interactions well. So I compare different QM approaches, that how well do they perform for protein glycan complexes. And I also use a combination of those GB models. So what combination of QM and GB model would give the best correlation when we compare from the experimental value? And you could see that we get the values that we get from model, let's say PM6, which is very quick, very fast to run, is exactly same like what we get from DFT approaches. So there's no point of kind of spending weeks to run the calculations on using DFT. You could simply run quickly PM6 and get the numbers, which are almost same. And you could also see that some of the models really poorly perform, so probably there's no point using them. And the second approach, second system that I applied this approach was a lectin that's called PNA, peanut agglutinin, and this lectin binds to the galactose. But our collaborator in Japan, they made some mutations in this sugar binding domain, and most of the mutations were to arginine. And upon these mutations, this lectin loses the binding activity for the galactose, but it becomes highly specific for heparins, which is like sulfated glycans. They have a lot of sulfate groups. So you could see that the wild type binds with different glycans, but it doesn't bind with the heparin, but the mutant binds with the heparin, and we try to explore that how this happens. And using those MMGBSA calculations, I could explain that, okay, what is the reason that why it loses the binding affinity for the galactose, and it becomes really specific, and the mutant one becomes specific for different heparin molecules, and we explain the molecular mechanism behind it. So that was the very first approach, the MMGBSA, and let's switch to the TI, thermodynamic integration approach, that's very close to my heart, because I spent a lot of time working on this approach and making it work for the glycans. And this is one of the most advanced techniques that we have now to get most accurate free energy calculations number. All of us are aware of thermodynamic cycle. So let's say you have a ligand and you make some modification, this yellow part into the ligand, and you want to see that how much would be the change in the binding affinity if you make some change in the ligand. How we handle in the calculation is that we know the binding, let's say we know the binding affinity of ligand A with the protein, and then we know binding affinity of ligand B with the protein. Or we don't know, actually, we want to calculate that. So instead of running these two simulations, we run these two simulations. So we try to modify this ligand from A to B in solution when it's not bound to the protein, and then we modify the same ligand in the bound state in the protein. And once we can calculate these two numbers, we can get the difference of binding energy from this thermodynamic cycle, because change in the binding energy along the cycle will always be zero. And that's how we get, so the difference of these two, bound state minus free state, would be change in the binding energy. How it is done in computation is that you start a simulation with the ligand X, and you end up with you end your simulation with ligand Y. And during the simulation, you have multiple windows where, let's say this when lambda is 0, it's totally X, and when lambda is 1, it's Y, and when lambda is, let's say, 0.01, it's 10% X, 90% X and 10% Y, and that's how it increases. It's a heavily computationally demanding approach, and even like calculating one modification for a small protein could take 10,000 CPU hours, but the predictions are very accurate. I'll just quickly explain everything that I was telling before, that if we want to see that how more or less tightly does the ligand X bind with the protein, this could be calculated using this thermodynamic cycle. So we looked at the change in the binding affinity of multiple sugars molecules when we transform this fucose into different molecules. So let's say transforming fucose into the mannose, into fucose, it's a methyl fucose into fucose, the difference would be only this methyl group. That's that one gap present here. Fucose into galactose, into fructose, rhamnose, into D-galactose, L-rhamnose, and I will talk about the intermediate later on. And these are the results that we get. So here I'm having, three bars. The first is the correlation between the experimental and the calculated free energy. And PI values are kind of indicator that if you pull out some of the ligand molecules, still how much correlation do you get? And mean unsigned error is the error between calculated and the experimental binding energy. These numbers, LI, is from my different paper that I had published, and these are different docking scoring functions that I had published before, but these two numbers are from using TI calculations. And you could see we get the best correlation between calculated and experimental values using TI approach. And these two bars, they just differ in the force field that we use. So in the first one, we use general Amber force field that's used for organic molecules. And in the second one, we use a glycan force field, that's a carbohydrate specific force field. And you could see that there's a big difference in between the general Amber force field and carbohydrate specific force. So we recommend that instead, sometimes we just don't care about the force field, and even if we have a sugar, we just treat as an organic molecule and general ligand, and we try to run the calculations using that. That's not a good idea. Rather, we should spend time in parameterizing it, and we should get, once we get the glycan parameters, we should use those, because you see that the difference in the error between these two programs. So that's the best to go ahead with. Often it happens that we are not able to perturbate one molecule into second molecule directly because of the steric classes. Once we are mixing two potentials, so you will get really unstable state at some lambda values. That is, you could see that we are getting different values. These are some of the examples. In that case, what happens? What I find it out that we could choose an intermediate, and so from one state A2 we could go to an intermediate, and from intermediate we could go to state B, and as long as our initial in the state they are same, intermediate doesn't matter. And by using that, we could choose an intermediate from going A to C. Let's see, we went to an intermediate D and then D to C. And we were able to fix this problem. Let's see, when we are going from A to B, A to C, there were really certain instabilities. But you go from A to B, the curve is smooth, and then you go from B to C, the curve is smooth. You just add up both of those. I'll quickly talk about that. How did I use this TI approach to make some modifications in the CD22 inhibitor, which is a very important receptor molecule to control B cell activation. When I started working on this project in 2016, there was no crystal structure available, and I had a hard time in trying to understand what's happening here, because every time we will model a structure, we were modeling this tryptophan residue, which was interacting with the sugar, because these siglets, there are different other structure from this family, and they all have this tryptophan into this conformation. And I was always getting a different conformation in the modeling, and it was really difficult to see that what's happening, why we are getting this one. But later, when the structure came out, before we could publish this data, we could find that what I was getting from modeling is actually correct, and the structure that we had modeled was almost accurate. There were just some differences in this region. So for this inhibitor, for this receptor, it binds to this NUAC, which is acetalic acid, and there are different analogs of this NUAC. So people have made modification in this position, in the C2, in this 4 position, and 3 position mainly. I did some TI calculations, and we proposed these three modifications at 4th position. So for these modifications, you could see that this first bar is the experimental change in the binding energy, and then these 2 bars are the calculated change in the binding energy that I had predicted using 2 different those TI approaches. First is M bar, and the second one is using TI. So in our cases, we got some C3 and C4 modifications that improved affinity, and then those molecules are still being synthesized, and they would be tested experimentally. And the last, I think it's part is that I'm going to talk about is that not only the binding for energetical reasons, we also work on predicting the structure of the proteins which binds with the glycans, or the enzymes that modify glycans, like those which play a role in glycosylation. So PGF4 is one of the enzymes that modifies this glycolipid GPI anchor. Some of us know that some proteins, they use this GPI anchor to attach themselves to the cell membrane. And there are different sugars here. And this PGF4 was the enzyme that was identified by one of our collaborators in Japan, and it modifies this GalNAc residues, which is N-acetylgalactosamine. They identified it experimentally, and they wanted to know that what could be the structure of this enzyme, and how does it look like, and how the catalysis happens. So I modeled the structure of this enzyme, and we came up with a model that has 3 disulfide bonds and the transmembrane region, and probably the UDP glucanac comes from this side, and this is a GPI binding site where the GPI would bind, a reaction would happen, and then it releases like this. Our collaborator, when they came to us, they said that there are 4 disulfide bonds, and they're certain about it. And when I modeled the structure, I could see that there are 3 disulfide bonds, but the 4th one that they were expecting was between a cysteine residue somewhere here and the cysteine residue here. And if you form a try to form a disulfide bond between these two when you are modeling, you would just close this cavity. And this didn't make sense to me. So we had 6 months of time arguing that. I was saying probably no, probably there's no disulfide bond. They were saying no, they have a data, there's a disulfide bond. And then after a few months, they said that, okay, this has to be tested. And they went on to the lab and they did mass spectrometry to make fragments of the GPI sequence, and then they do some experiments, and they made it clear, did the mutations and just to see if this is stable or not, and then they could verify that, okay, actually there's no disulfide bond that they were expecting initially. So with this, I would like to thank all the people that I have worked with. Mainly they were my supervisor, but especially Professor Johan Ekvist and Julian for this TI work, because I worked in his lab in University of Edinburgh when I was optimizing this free energy perturbation protocol, and I implemented this in his software. And with this, I would end my talk. If you have any questions, feel free to ask. And if you don't get enough time to ask the question, you could just see my email at the very front page. I would change it to this later. Now, you could just drop me an email about that. So thanks for paying attention.
Thank you, Dr. Mishra. It was a very nice talk. I don't see if people have actually posted any questions. Maybe they'll try to directly contact you.
Okay, so there's one question, I think.
Yeah.
You have validated the in silico results with experimental data.
Experimental data.
What's
What
the
is
correct?
the question?
That's what I was showing, that the correlation. Where are my slides? Oh. So these are the correlations that you could see here. I was showing that how well it correlates, like the experimental and the calculated value correlates in MMGBSA. And in the case of TI calculations, so this bar is the correlation between calculated and the experimental value. So we would say that we got it above 0.9, and that's Pearson correlation coefficient. So that's very good for computations, because anytime if we are plus minus 1 kcal per mole from the experimental binding free energy, I think that's quite successful prediction. Is there any other question?
That is the only question we had. I think people try to, they're trying to cope, understand the lot of calculations you have done and a lot of in silico data. So it's a bit tough. And I mean, for wet lab people also, it is tough. But it was very nice talk, and I could see that your experimental data, wet lab, and they're matching quite nicely, as you shown that your Japanese collaborator data and your data, you were from the beginning you were arguing that no, there is no disulfide bond, and then the wet lab they confirmed yes, there is no disulfide bond as you predicted. Oh, very good work, very good. Very
Thanks.
nice.
You know, our general understanding is that when there is some experimental data, everyone will trust this data except that person who has done the experiment. But about the calculations is that when you have some calculated data, no one will trust it except the person who has done the computations. So that's something. Sometimes you do the computations and like you feel like no, that there should be this or not. And that's how we succeeded in this PF4 project.
Yeah, but
Okay,
anyway, it was very
thanks a lot.
convincing. Thank you, Dr. Mishra. It was very nice presentation.
Thanks a
Thank
lot.
you. Thank you.
Take care.
Now we move on to the next part of our session today. It's oral presentation, and I have here a list people who missed yesterday. They couldn't present for some reason. I call upon B. Surumi to go for presentation. You have total 7 to 8 minutes, including your talk as well as your some questions which can be asked by the people who are attending this conference. B. Surmi, please. You can share your slides. Miss Surumi, please start your presentation. We can see your slide, but we cannot hear you. No, she's. Surumi, can you please start your presentation? Surmi, are you ready with your presentation, or we should move on to the next presenter? We cannot hear you. We can see your slides, but we are not able to. No.
Right now you can hear me?
Yeah, now we can hear you. Please.
Okay. Sorry for that. Good afternoon, everyone. I'm Surumi from Kerala University. My topic of presentation is expression analysis of steroid receptor RNA activator long non-coding RNA during 3T3L1 pre-adipocyte differentiation. As an introduction, we all know that obesity is considered as a killer lifestyle disease and it has become a global epidemic. And according to WHO, 1.2 billion people are considered overweight worldwide. India is just behind US and China among top 10 countries with highest number of obese people. Obesity is a condition of abnormal or excessive fat accumulation that is harmful for human health. Adipose tissue is made up of adipocyte, and the process of formation of adipocytes from pre-adipocyte is called adipogenesis, and the adipose tissue is mainly of two types, white and brown adipose tissue. And the white adipose tissue is responsible for fat metabolism and storage. Bat or brown adipose tissue is responsible for non-shivering thermogenesis. Adipogenesis pathway is studied because of its contributions to obesity and diabetes. The obesity is characterized by mainly two conditions, that is increase in size, hypertrophy, or increase in number of adipocyte. Why we selected LNC RNA in adipogenesis? Because of the increase in prevalence of obesity, and this process is intensively studied due to the availability of good cell culture models that are not possible in other studies. Then understanding the process of adipogenesis is very important, that it is leading to obesity. The adipogenesis is mainly regulated by many genes, transcription factors, and cofactors. Recently, many remarkable regulators came into existence like long non-coding RNAs. These long non-coding RNAs are non-coding RNAs that are having a size range greater than 200 base pair, and they have many features similar to mRNA, like they have a poly A tail 5' cap, they are multi-exonic, but they does not code for any protein. They are RNase polymerase 2 or 3 transcripts. They may or may not have a poly A tail. They are in the nuclear or cytosolic fractions. They are involved in main process of gene expression like epigenetic, transcriptional or post-transcriptional levels. The first recognized LNC RNA is imprinted H19. The objectives of the study is to check whether the SR-LNC RNA is involved in adipogenesis or not, or check whether the pattern of expression is altered during the adipocyte differentiation. Work outline involves 3T3L1 cell culturing, then adipocyte differentiation using established method, and check the adipocyte differentiation by oil Rnas staining, then RNA isolation during different phases of adipocyte differentiation by trisol method, checking of RNA quality by agarose gel electrophoresis and spectrophotometric quantification, cDNA synthesis by Verso kit method, quantitative real-time PCR based on cybergreen, and the fold chain calculation. These are the results. 3T3L1 cells after 6 hours and 24 hours in culture. These are the morphological analysis of the cells during the different phases of differentiation. These are oil Rnas staining showing fat droplets during different phases of differentiation. RNA samples during different phases of differentiation. The spectrophotometric quantification values of RNA samples during different phases of differentiation. cDNA was also synthesized from this RNA. Then 3 reference genes were checked, 18S RNA, GAPDH, and beta-actin were checked, and from which 18S RNA was selected as the reference gene. These primers were taken from literature. These are the real-time results showing melt curve, melt peak, and amplification curve of 18S RNA and SRA. From the real-time experiment, it has been found that the expression of sRNA is more toward the adipocyte phase when compared to pre-adipocyte. It is having a positive regulation during the adipogenesis process. The real-time samples were run on 1% agarose gel to check whether we are getting a specific product, and from which we got that it is having a single band and the product formation is specific. These are the results of 18S RNA. Next, we have real-time products run on gel of SRA. As a summary, we have found that 3T3L1 cells were differentiated effectively. Good quality RNA was synthesized, cDNA was synthesized, and by real-time PCR, we could find that a positive regulation was shown by SRA-LNC RNA more towards the adipocyte phase. And this unique pattern of SRA indicate that it is having a clear role in obesity and adipogenesis. And a functional study of this SRA-LNC RNA could help us to develop newer strategies for combating obesity. Thank you.
Thank you, Ms. Surmi. It is open for discussion. The presentation is open for discussion. If you have any question, people can ask. They can type and I can ask on their behalf. Any question from audience? Okay, thank you, Ms. Surmi. It seems there is no question, so we move on. Thanks for your presentation. It was a nice presentation. We move on next presentation by Sweta Minhas. Sweta Minhas, are you ready with your presentation?
Yes, sir.
So you can start your presentation now, please.
Yes, sir. In a while. Good evening, everyone. So, am I audible? Hello?
Yeah, you are audible. I think you are using your mobile phone for presentation.
Yes,
So
sir.
go for full screen display.
Okay, sir. So, is it visible now?
Yeah, now visible. Please.
So, the title of the study is Optimization of L-Asparaginase obtained from Enterobacter asperae using response surface methodology, isolated and characterized from black gram-basal spherical samples. Now, L-asparaginase is an amido hydrolase that catalyzes the breakdown of asparagine into aspartic acid and ammonia. The enzyme has an important has served as an important chemotherapeutic agent, and it was the first enzyme with anti-leukemic activity to be studied intensively in human beings. The basic mechanism of action underlying the chemotherapeutic role of this enzyme is that it causes a nutritional deprivation to the cancer cells, since the L-asparagine that is circulating inside the human blood that provides a nutritional source to the cancer cells. When this L-asparaginase is injected inside the body, it converts the circulating asparagine into aspartic acid, which causes a nutritional deprivation to the cancer cells, thus leading to the cell death. Uh, Therefore, considering the role of enzyme in chemotherapy, the study aimed to isolate a potential L-asparaginase producing culture and optimizing different parameters for large-scale production of the enzyme. The methodology followed for this study, it included isolation and screening of asparaginase producing culture from black gram rhizospheric soil samples. First of all, the samples were collected from Rajouri region of J&K. Then the samples were serially diluted. After that, they were spread plated on nutrient agar plates to isolate a pure culture. After the isolation of pure culture, the screening was done. The screening was done both qualitatively as well as quantitatively. For the qualitative screening test, we used a rapid plate assay method where 0.1% of phenol red indicator solution along with 1% asparagine was added to the nutrient growth media, which will result with a change in color from neutral or yellow to pink, which is due to the release of ammonia because of the enzymatic action, thus confirming the presence of L-asparaginase positive culture in our growth media. Then for the quantitative determination of this enzyme, we used Nestlerization method, where Nestler's reagent was added to the assay reaction, which would again result into the change of color, and the color intensity was then measured spectrophotometrically at 436 nanometer. Then the culture was characterized using biochemical characterization test as well as 16S ribosomal RNA sequencing method. Then different parameters were optimized for the large scale production of enzyme. Different parameters that were optimized, that were incubation time, different ranges of temperature were used. Then different pH ranges were also used for the optimization. Different concentrations of the asparagine were also tested. Then percent inoculum was also varied to optimize the proper inoculum size for the growth of the for achieving enzymatic activity. Then different ranges of RPM and different types of media were also used, wherein the variation was given to the carbohydrate source that was supplemented in the media. Along with that, some pH ranges were also varied, and various other salt ingredients were also varied, and so that 11 different media that can be used for the optimization of a specific media for the asparaginase production. Then here, on the basis of optimization results that were obtained, the major influential factors contributing to asparaginase production were taken, and to identify the best combinations of these factors, 15 sets of experiments were conducted using response surface methodology, and the experimental data obtained was then used to determine optimum point of process parameters using a box-Penkin design. Now coming to the results of the study. Here, figure 1 shows different bacterial colonies that were isolated from the collected soil sample. And here in figure 2, we can see the asparaginase qualitative screening results, wherein the change in color, this pink colored zone, which is formed around the culture growth, indicates an asparaginase positive culture, which was isolated from the soil sample. Then a quantitative determination of the enzymatic activity was done, wherein an ammonium sulfate standard graph was taken, and the enzymatic activity, which came out to be 9.15 units per ml, was calculated against the standard graph. And then 16S ribosomal RNA sequencing results of the isolated culture. On the basis of the RNA sequence that was derived, a phylogenetic tree was constructed, which showed a relationship between R16P1 strain and various other Enterobacter species that were related. So based on the sequence homology results, this Enterobacter, this particular culture was characterized as Enterobacter asperae. Now here it is a Pareto chart which shows effect of different variables that were used for the study. So the most influential factors here were media, as there was an increase in activity from 9 units to 30.6 units per ml in this M9 media, which is a maltose supplemented media that was used for the study. Along with that, a concentration of asparagine, which is around 1%, gives maximum enzyme activity. Then other factors were inoculum, pH, RPM, temperature, and incubation time. So here the interaction between the process variable design is represented using 3D surface plots, where this figure shows response surface plots obtained as a function of RPM, temperature, and inoculum. Since RPM, temperature, and inoculum size were the three factors taken for the interactive study further using a box pan design using response surface methodology. So this figure indicates here in this figure AB, it shows an interactive effect of temperature and RPM, keeping inoculum size as constant. And then similarly in this figure B, see here RPM and inoculum are studied together, keeping temperature as a constant factor. And in the third figure, which is designated as AC, temperature and inoculum effect was studied combined, taking RPM as a constant factor. Now here in table 2, it shows analysis of variance results for the experimental design, where p-value less than 0.05 indicated the significance of or model terms. In this case, temperature, RPM, and inoculum size, and the combination of these three, which is RPM, temperature, and inoculum size were significant models. The adequacy of results analyzed using this ANOVA and R-square shows a good fit between predicted and observed responses, thus implying the reliability of the model. So coming to the conclusion of this study, a potential L-asparaginase producing culture was isolated from the soil sample of Blackgram. L-asparaginase production by isolated culture was initially optimized using one factor at a time. Then to study the interactive effect of the most influential factors, a box Pancon design was used to optimize the interactive effect of different parameters. The significant interaction between RPM, temperature, and inoculum was observed using the response surface and contour plots. Response surface methodology, it proved to be a significant tool for optimizing different parameter effects, resulting in a 5.5-fold increase in enzyme activity, which was from 10.24 units per ml to 40.09 units per ml. A high similarity between the experimental and predicted values as given in the table number 1 in this study. It directly reflected the applicability and accuracy of design to optimize different variables for asparaginase production. Thank you.
Thank you, Ms. Manhas. Now it is open for discussion, and questions are welcome. I think there is one. I think there is no question audiences are asking, but I have a question for you, Ms. Manhas.
Yes, sir.
You said this you are going to use for L-asparaginase for depriving cancer cells, right?
Yes, sir. Yes,
Basically,
sir. Yes, sir. Yes, sir.
now this is a bacterial enzyme. How you
Yes,
use
sir.
it?
Sorry, sir. How you?
How, I mean, what you have in your mind that how you'll use this bacterial enzyme for in say
Yes, sir. Thanks. Yes, sir. So actually, the enzyme has been used already for the chemotherapeutic treatment of cancer patients. And it is a huge success in the field of acute lymphoblastic leukemia. But at present, actually, there are like only two potential sources of the enzyme which are being used. Plus, those two sources are associated with certain hypersensitive responses. So the main aim is to look for a new source of the enzyme, which could have a less hypersensitive reaction as compared to the already being used sources of the enzyme, plus large scale production of enzyme if that can be brought up with this new source. But it is being used for the treatment of acute lymphoblastic leukemia in the form of intramuscular or intravenous injections already, sir.
Yeah, that partially answers my question, because you're telling hypersensitivity, it is showing it's going to be a foreign molecule.
Basically, this paragenase that is currently being used for this chemotherapeutic treatment, that is obtained from E. coli and Erwinia species only. So these two particular species are showing like some hypersensitive reactions also along with the therapeutic effects. That is why I was looking for something that could have a less hypersensitive response.
Thank you. Nice work. You're looking
Thank
from
you.
the very beginning. Thank you.
Yes, sir. Thank you so much, sir.
Now we move on to the presentations which you are supposed to have held today. So first is Gidirappa. Am I correctly pronouncing your name?
Yeah, yes, sir. Yes, sir.
Okay, please, floor is yours.
Yeah, thank you, sir. So, good evening to all. Am I audible?
Yeah, you are audible. Your slides are very clearly presented. So you can go ahead, please.
Okay. Thank you, sir. Thank you. Myself, Gidirappa from Department of Chemistry, VSK University, Ballari. So today my talk of presentation is based on managed reaction derived phthalocyanine polymer for electrochemical detection of salicylic acid. So before going to the main presentation, we have to I would like to give some brief introduction about phthalocyanine and redox active molecules. So phthalocyanine, basically it is a N4 macrocycles, and it is a very important biorelated and material science application based compound, and the properties of these N4 macrocycles can be tuned by tailoring or monitoring the peripheral benzene rings with various substituents, various chemical substituents, and these properties have been exploited in different applications from researchers in all over the world. So a lot of efforts have been put to synthesize various substituent metal phthalocyanines and study their properties also in different application way. So even though uh, and other redox active molecules are basically the metal phthalocyanine is a redox active molecule. It can compete with quinone redox couple, which is a redox active molecule, and polyaniline, and Prussian blue, as well as graphene and fullerene. So my talk highlights is like the precursor. We synthesized metal complex, that is polymer. The precursor compound is prepared by the managed reaction. It is the uh, managed derivative structure, and then managed base was attached to the 4-nitrothalonitrile to get polymeric phthalocyanine, and the synthesized phthalocyanine were characterized by different various uh, analytical techniques and electrochemical techniques, and this polymer was drop coated on the bare GC carbon electrode for the detection of salicylic acid using cyclic voltammetry, differential pulse voltammetry, and chronoamperometry. The composite hybrid polymer on the GCE exhibited a high sensitivity and reproducibility for the detection of salicylic acid. Though my scheme of synthesis is like first step. In first step, we would synthesize the managed base ligand. Why we went for the managed base? It is the managed reaction involves three-component nucleophilic addition reaction forming carbon-carbon bond, which is fundamentally important in organic synthesis, and it is highly effective in the preparation of enantiomerically enriched compounds like polymers, resins, detergent additives, surface active agents. In that field, the managed-based ligand were used, and this is the first attempt which has been made to synthesis managed-based substituent phthalocyanin by using this managed reaction. So here first the 4-hydroxyacetophenone, ammonia, and vanillin reacts with each other in the presence of low boiling solvent, that is ethanol, and citric acid as the catalyst. So we'll get the we got the product one, that is a managed based ligand. Here we have OH, so which is react with 4-nitrothalonitrile in the presence of base, that is Ka2CO3, and solvent dimethyl formaldehyde. So we'll get the compound 2, that is nitrile based ligand. So from this nitrile based ligand, we synthesized the polymer, that is managed based phthalocyanin polymer, by adding cobalt chloride. So firstly we can I told we can tune the phthalocyanin by various metals as well as various peripheral substituents. So after synthesizing this polymer, we characterized with different techniques, that is physiochemical technique. First we have we characterized the polymer by UV visible spectra. So in UV visible spectra, it is shown two intensive peaks, that is one for 325, another one for 670. So that is 670 nanometer corresponds to the Q band, which is arises due to the pi star transition, and this 325 nanometer, which is arises due to the n to pi star transition. And if you go to the IR spectra, in first step we don't have any cyanide peak, and in the second step we have the strong absorption peak at 2200. It shows our ligand is prepared in the good way. And final step, this is the synthesis polymer phthalocyanin IR spectra. So here we can say our polymer has been synthesized properly. So our phthalocyanin polymer were characterized by powder XRD. So here we cannot see any intensive peak. So from this result, we can say that our phthalocyanin compound is amorphous in nature, and this is the TGA spectra for thermal stability studies. So from this figure, we can say our synthesized polymer is stable up to 350 degrees Celsius. Then it is decreased, it is slowly decreased due to glassy nature of that polymer. So if you went to the if you see the electrochemical measurements, so before going to the electrochemical measurements, the electrochemical behavior of synthesized polymer was studied by dissolving 5 milligram of the phthalocyanin polymer in 10 ml of aprotic DMSO, that is dimethyl sulfoxide, having 0.1 molar TBAP, that is for conductive increase the conductivity of the solution. So here we can say that there are this cyclic voltamogram of the PC solution displays two redox couples and can be assigned for two ring based, that is the cobalt will get oxidized and reduced to the +2 to +1. Then the other peak for the cobalt metal. Then this is the ferricyanide system for knowing the charge transfer studies. The charge transfer studies behavior were characterized using a Redox probe, that is a ferroferrous cyanide system. That we can see that here the peak to peak separation, that is delta E value, is about 179 for the bare electrode, and 229 for the polymeric phthalocyanine. So from this we can say that this may be um, the complex nature is thickness of the film and conductivity of the composite. So then, then finally uh, we examined the polymeric material in the electrochemical system for the detection of the salicylic acid. Why we went for the detection of phthalocyanine? Because of people is phthalocyanine, basically it is a beta hydroxyl acid derivative found in appreciable amount in the foodstuff derived from the plants, as well as aspirin is salicylic acid is a basic product of aspirin. So nowadays people who consume excess dose of aspirin may suffer from the different diseases like atherosclerosis, colorectal, and other soft tissue cancers.
Mr.
Yes.
Gidirappa, can you hurry up? We are running short of time.
Yeah. Within 2 minutes, I will conclude my talk, sir. Thank you. Sir.
Quickly. Okay.
Yeah. Quickly. So because of this reason, it has to be detected and monitored regularly in the system. So this is the uh, sensing part. If we on adding the salicylic acid analyte to the sensor, it will increase the peak potential. And this is the mechanism. Basically it involves two electron process, and from this sensing part we got the limit of detection value, that is 2.8 micromolar, and linear range between 10 to 100 micromolar. So we done the same for differential pulse photometry using the polymeric phthalocyanine material, and upon adding the salicylic acid analyte, the increase in peak current, it shows our phthalocyanine material is can able to sense the salicylic acid. So from this DPV, we got LOD 2 micromolar and linear range from 5 to 90 micromolar. From chronoamperometry measurements, we got the LOD 0.12 micromolar and linear range from 0.5 to 4.5 micromolar. So interference study shows that the stability or specificity of our phthalocyanine complex. So from all these results, we can conclude our composite material, that is phthalocyanine, as well doped with Kedjan black, that is carbon nanoparticle, is displayed better electrochemical signal for salicylic acid, which may be due to the presence of polymer and carbon nanoparticle. So the developed sensor has a possible potential application in the detection of micromolar concentration of the salicylic acid in the real sample. So the proposed electrode can show better LOD sensitivity, stability, and reproducibility compared to the other electrodes which are in the literature. This is the final TOC, and thank you for your attention.
Thank you, Mr. Gidirappa.
Yeah, sir. Okay, sir.
Thank you. Now we move on, and next presentation will be.
Is there any questions?
No, I'm afraid there is no question.
Okay, sir. Okay.
Okay. Thank you for your
Yes.
concern. Thank you.
Yes.
Now we move on, and next presentation will be by Mr. Vinayak Rudra. Mr. Vinayak.
Yes, sir, I am here.
Okay, please. And please stay in the time limit that you are
Okay,
given.
okay, sir. Okay, sir. Sir, is my screen visible?
Yeah.
Sir, is my screen?
Yeah. Yeah, it is visible, but rotate your phone so we can see it clearly.
Okay, sir. Now.
Yeah, please.
Okay, sir. Okay, sir. Thank you. Okay. Good evening, all in one. Myself, Rudra Vinayak. I am a postgraduate alumni of Sri Konda Lakshman Telangana State Horticulture University, Hyderabad. I am here to present my key findings of my master's research that I have undergone a conducting a baseline study on barcoding data of velvet bean. Velvet bean is I collected my germplasm as a wild cultivar in this area. You can see here in India from Telangana itself. Coming to the crop velvet bean. Velvet bean is a leguminous crop of vegetable origin. In medieval periods, it is widely used as one of the green vegetable crop by number of tribes all across the globe. But due to progress of time and development, now it is present. It is only confined and cultivated as medicinal plant due to its immense medicinal properties. My objective is to rejoin this medicinal plant as a cultivable vegetable plant. So I undertook this study to read about the genetic barcoding data of this plant. I collected this plant in Nizamabad district of northern Telangana region. Later I extracted the DNA by using cetyl trimethyl ammonium bromide method, that is called CTAB. Again I determined the quality of DNA by agar-based gel forces at 280 nanometers using nanodrop ND1000 spectrophotometer. I undertook an exosap IT treatment using two hydrolytic enzymes. I run my DNA data in NCBI BLAST. So I acquired a query of 100% in all the data regarding earlier mucuna pruriens varieties in the NCBI BLAST program. So you can see the query cover here, all 100% with all mucuna pruriens varieties, and it is 98% with mucuna pogae. Yes. Next, the mean interspecific pairwise distance among the different species of mucuna was nearly negligible with the mucuna pruriens group, and it is 0.006 with mucuna pogae and 0.029 with Cochianthus glacialis. These are the photos of gelator forces regarding my DNA extraction, and I run with the NCBI BLAST program. This is the BLAST I got in that NCBI data. They have assigned a accession number that named as MN381025.1. Now this plant is named on my name in the NCBI database. With these data, we have constructed this tree by using neighbor joining method under distance model of 3 Kimura parameter that distinguishes adenine thymine to guanine cytosine transversion with adenine cytosine guanine thymine uses MCL approach, that is maximum composite likelihood compost, and this robustness is evaluated by bootstrapping with 1000 replicates. Here this is the P1 is my plant, and those are all the clades. Subclad 1 is totally on par with my plant data, and subclad 2 that's showing mucuna pogae is the immediate perennial ancestor of my plant, and the Cochianthus glacialis is the outgroup of the mucuna pruriens. By this we can my key findings of this research was the nucleotide sequence of mucuna pruriens presents 99.7% identity with the mucuna cochinensis and 99.38% identity with mucuna pogae. Sequence currently available in the nucleotide data of NCBI. The immediate perennial ancestor to annual varieties of mucuna pruriens is mucuna pogae, and all annual varieties of genus mucuna in India belongs to the species mucuna pruriens. MATK could be the DNA barcode to resolve the taxonomic boundaries of closely related mucuna species which share close phenotypic features. My future line of work is focused on the progress is expected to pave a way for successful integration of molecular markers in breeding, starting from development of saturated linkage maps and identification markers, QTL linked to L-dopa trichomes, mucinal content, and other economic traits. Although mucina has been underutilized for many years, application of genetic barcoding technology provides new opportunities to identify and select better adapted genotypes with high yield, improved food and feed value, and reduce anti-nutritional factors when ultimate goal is medicinal and greater L-dopa content. Thank you.
Thank you. Thank you for your presentation, Mr. Vinayak. It was a nice presentation. So you are working on barcoding of this mucuna.
Yes, sir.
It's basically a medicinal plant. It is used for a lot of actually Ayurvedic medicine, right?
Yeah, yes, sir. Yes, sir.
Okay, good work. Next, we move on to the next presentation of the day. It is by Yukeshwaran L. Mr. Yukeshwaran, are you ready with the presentation?
Yes, sir, I'm ready, sir.
Yeah, please. The floor is yours.
అంతే అన్నట్టు మాకు
Sir,
నేమ్
whether my
వాడలేదు.
screen is visible, sir.
Yeah, slides are visible.
Post a presentation.
Yeah, please make it full screen presentation and then start presentation. And please stay in time limits. We are already late.
Okay, sir.
Yeah.
So may I explain, sir?
Sorry?
Sir, may I start my presentation?
Yeah, please do start.
Okay, sir. Okay, sir. Good evening to everyone present here. I'm Yugeshwaran Loganathan from Banariam Institute of Technology, Tamil Nadu. I'm going to present my work entitled Immunoinformatics Aided Multi-Epitope Based Vaccine Design Against Marburg Virus. In our work, we focused to design the epitope-based vaccine to treat the virus, the Marburg virus, which causes a highly virulent disease and a hemorrhagic fever.
Mr.
Hello, sir.
Can you increase the size of your presentation a bit? Yeah.
Okay, sir. In our work, we have focused to design the epitope-based vaccine to treat the virus, the Marburg virus that causes highly virulent disease, hemorrhagic fever. The host of this virus is an African fruit bat. Merck has disconnected the designing of RSV virus and also noted that the Soviet Union included this MOV as their biological weapon. And here I represented the geographical distribution of this Marburg virus. The red color area denoted that the reported cases is greater than 50 per year, and orange denoted the 5 to 50 cases, and yellow shows the astrological evidence that the people affected from this virus. The lightish yellow region shows that the presence of this virus host. So the chance may be high for the evolution of this virus come to result in 10% of fatality. Okay, coming to the methodology that we designed the epitope vaccine. First, we extract our proteins using a UniProt. Then we checked the similarity index using a BLAST tool and multiple sequence alignment using a cluster omega. And we find our B cell epitopes using a ABC PRED. And we have screened our epitopes by checking their antigenicity, allergenicity, solubility, and also stability. We also checked using online servers, which I mentioned here, like Vaxigen, Allergen, Protoprom, like that. Then we find a T cell epitope, and we used a final screened epitope to design the peptide vaccine. And this is a Marburg virus structure. From this, we have taken the proteins. Then we have checked their similarity index. We have compared whether the genome sequence showing any similarity with the human genome. For this, we used the NCBI BLAST tool. We came to found that the similarity is less than 40. So we came to found the 3 proteins. These are the 3 proteins: minor nucleoprotein and membrane associated protein and envelope glycoprotein, which have less than 40 similarity. Multiple sequence analysis plays a vital role in this part. We have screened epitope which is common to all strain of Marburg virus by using cluster omega tool. We have taken these 3 proteins and and envelope. This both have these three proteins have 83 epitopes. Out of we have applied screening parameters to screen these 83 epitopes, and final we have get only one epitope. This is a 3D model of our screened epitope, which is get from the nucleocapsid protein. And these are the MHC alleles for T cell epitopes of replicase protein, which resulted is a greater binding with the T cell. And the screened epitope, which satisfy all condition, is isolated from the minor nucleoprotein. And these are the parameters of the particular epitope, which resulted in antigenicity at score of 0.7, and the epitope is not an allergen, and it is more stable with the index value of -9.34, and it is non-toxic in nature. So the higher solubility with the hydrophilicity of 0.45, and the epitope is not transmembrane and signal peptide. Here we have found the Ramachandran plot for of the particular epitope. Here is the graph. You can see that it is zoom. In this graph, you can see that the major region is present in alpha helix and beta sheet, which results in 100% of favored and allowed region. The particular epitope is isolated from nucleic replicase protein, have the following properties like molecular weight of 1743.98 and charge value of 1.5 with a theoretical pi of 8.51. And I conclude that our epitope will be a greater part to design the peptide-based vaccine against the Marburg virus. And these are our references which we used for our research work. And I conclude my presentation, sir.
Thank you for your presentation.
Thank you, sir.
Lukeshwaran. So you are basically trying to look for some epitopes that can be used to develop vaccine for Marburg virus. Yeah, good work. Okay, next we now move on to
okay, thank you very much, sir.
Next presentation. This will be done by Mr. Jaydev Sharma. Mr. Sharma, are you here? Mr. Sharma? We can see you, but your voice is not really audible. It's very weak. Barely, it's very weak. Hello. It's very feeble. Your voice is not really audible. No, not now. Okay, can we move on to next presentation by someone else, and then we come back to you. In the meantime, you check your connections. Himangi Ranade, are you ready with your presentation? Can you please start presentation?
Sure, sir.
Please.
Is the screen visible?
Your screen is visible and your voice is very clear. Please start
Okay,
presentation.
sir. Thank you, sir.
Thank you.
Good evening to one and all present here. My name is Himangi Ranade, and I am a PhD scholar working in Amity University, Rajasthan, under the guidance of Dr. Manali Datta. I'm here to present on our work, Conceptualization of Handy Water Treatment Device Targeted Against Pseudomonas. Starting with my presentation, I would like to recollect to all the listeners that in today's afternoon talk by Dr. Vishwanath Tiwari sir from Central University, Rajasthan, he gave a very informative lecture on escape pathogens. One of that escape pathogen is Pseudomonas aeruginosa, which is a leading cause of around 30% of hospital-acquired infections. So the main reason, one, which was because of which we chose this pathogen, is that it is a nosocomial infection causing pathogen. Second is that it is an opportunistic pathogen. It targets individuals with weak or immunocompromised system. It is a very easy to grow pathogen which can be transmitted from one person to another. Even it grows on moist surfaces such as sinks, ventilators, and catheters. It can grow in contaminated water, uh, distilled water, or even potable drinking water. So, according to WHO, it is a priority pathogen, and according to the Center for Disease Control, it is considered as a serious threat. Moving on, this is our system that we have devised. It is a novel, innovative method in which we have used filter paper strips. We have used two molecules, which we have named attractant and annealant, based on their property to attract and inhibit or kill Pseudomonas, respectively. So we have taken the strips, we have dipped them in that particular molecular solution, and as the solution is absorbed into the strips, that strip is named as an attractant, and the other strip which is dipped in an annealant solution, the solution is absorbed onto the strip, and that strip is named as annealant strip. We have joined it and made it into a whole system. Now, as a device, when we are using this system, we dip the system in such a way that the attractant end is dipped inside the culture or the contaminated water, and the annealant end is present above it. Now, with the brief working, we normally dip the system and keep it for various incubation time. Let's say I say 1 hour of incubation in this current presentation as I am displaying. We measured various experimental data upon based on which we have generated the efficiency of our system. First, we took the bacterial load and we measured electrical conductivity of the culture after 1 hour of incubation. So after that 1 hour incubation, the electrical conductivity was seen to be increased. And this is what we had expected based upon the past research. Secondly, we were sure about the inhibitory effects of the particular annealant with the results that we took the strips after incubation with pseudomonal culture. We kept the strips on the agar plates, and after an overnight incubation, it was clearly visible that the pseudomonal growth was almost nil at the annealant end, and a bit of the growth was observed at the attractant end. Another way in which we confirmed the trapping of Pseudomonas because of the attraction of the attractant molecule was that we characterized it with the scanning electron microscopic analysis. We had the images where we could absolutely detect the bacteria which is trapped inside the porous nature of our filter paper. Another is we measured the percentage bacterial removal by taking the differences in the OD of our bacterial culture with respect to the increasing incubation time. These results are displayed on the slide, wherein at the left side, we can clearly visualize the STEM images with the bacteria trapped inside the porous nature of the filter paper. On the right hand side, it is clearly visible that the annealant end is not having even a single colony of bacteria. So that confirmed that our annealant is working very efficiently. This particular graph is showcasing two important things: the increasing electrical conductivity with respect to time, and the increase in bacterial removal percentage with respect to time. This was measured with the help of OD. So this is a device which we are proposing from our work, that we can generate a handy device which will be able to sanitize, and which will be used as a potential point of use water treatment device, wherein we have all the molecules which are naturally occurring, organic in nature, and are cheap, and we do not have any chemical which will cause any hazardous effect to the water which we are trying to treat. So it is environmental friendly, and it will provide as a green and a clean technology which can be used for water sanitization. These are our outputs which we have generated. we are planning a handy device. We have generated the system already, and the main advantage of the system is that it can be used as a point of use device. Another advantage is it is not dependent on temperature change, pH change, or any other factors are not uh, altering its efficiency in any manner. It can be a very cost effective product because all natural and organic and cheap materials are used, and it can be an energy efficient sanitization device without a doubt, because it is not requiring any sort of external energy. It does not require any skilled labor. It does not require any extra knowledge. It's just a handy device which should be dipped inside the water, and you will get a potable natural water without any contamination. So this is what we are aspiring, and we are trying to generate the proposed model. Thank you very much for your attention.
Mhm. Thank you, Ms. Ranade. It was a
Thank
nice
you, sir.
presentation. So I think I got you correct that you said you used all natural and organic molecules,
Yes, sir, certainly.
both for attracting these bacteria and also killing these.
Inhibiting. Yes, sir, yes, sir.
Okay. Did you check residual bacterial load in the water that you have treated using these
Yes, sir.
devices?
Yes, sir. About after 2 hours of incubation, we are getting more than 95% of removal efficiency.
Okay, and I think you will not disclose what material did you use
No,
for
sir.
these.
Actually, we have applied for a patent, sir. And as soon as we get a provisional, I'll certainly mail you those products, sir.
Okay, thank you. Nice presentation. I think there is no other question from audiences, so we'll move on to the next presentation. Thank you, Ms. Ranade.
Thank you so much, sir.
Next presentation will be by Wail Ali Muhammad Hadi. Mr.
Yes,
Hadi,
yes,
are you ready?
yes. I am audible.
Yes, you are audible.
Okay, okay. I will present now. Can you see my slides?
Your slides are visible, clearly visible, and you are audible. Nice.
Okay, thank you very much. Good evening, everyone. So I am Wael Ali Muhammad Hadi. I am from University of Kerala, Department of Biotechnology. My topic is isolation of L. asparaginase producing fungi from marine water. This is like poster presentation, so I will make it in brief. Introduction: L. asparaginase is an enzyme that catalyzes the L. asparagine hydrolysis into L-aspartate and ammonia. It is used as an anti-cancer medicine and in food industry to eliminate acrylamide. Acrylamide also it's considered like a cancer agent. Marine habitat is promising as a harsh environment in which organisms and their product gain a salient properties that needed for industrial application. The present study demonstrated the fungi isolated from marine water have the capability of producing L-asparaginase. Methods: it is divided to three measures. So first is color change based on the pH indicator. This is for the first screening, and this is used dye based for as three major we are used: phenol red and bromothymol blue and bromocresol purple. And this the change of the pH, the color can be identified. Second, Nistler assay, and this is detect the liberated ammonia as international unit, and one unit it's the release of one molecule of ammonia per minute at 37 degree. Third, identification using ITS 18S gene sequencing. We come to the result. Among 48 fungi, 6 species selected according to color intensity from the primary screening and Nistler assay. An isolation of the 18S RNA from this fungi, it is resulted for 1658 among all of the six organisms. So all of these six organisms were selected all together for the sequencing and identification. These organisms also deposited in NCBI bank for the after the phylogenetic tree and for another for further studies. Thank you for this.
Thank you, Mr. Ali Muhammad Hadi. So you have six fungal fungus isolated from this marine
Yes,
source, and all
yes,
are showing the enzymatic activity. Very good
yes.
work. Congrats. Now, I don't see anybody has asked any question from your presentation. So we'll move on to next presentation by Mr. Jaydev Sharma. Mr. Sharma, are you ready now? Mr. Jaydev Sharma?
Am I audible now?
Yeah, now we can hear you.
Okay, sir. I think there was some problem with my mic.
Please start your presentation.
Is it visible now?
Yeah, visible. Speak little loudly so everybody can hear you.
Okay. So, good evening, everyone. Topic of my presentation is about designing the inhibitors for COVID-19 main protease for their adamant analysis. And let's start. So, I'm basically a student from Center for Bioinformatics, Maharishi Dayanand University, Rohtak. I started my work on this particular protein because when I was searching about a druggable target for COVID-19, I found 25 druggable targets in entire proteome of the COVID-19. Among them, the protease was one of the most attractive target, as it could initially lead to the inhibition of the synthesis of various proteins from the polyprotein, as this protein mainly cut the polyprotein into many functional proteins. So for that, we we took the genome of near about 118 strains that are available on NCBI virus. We found that the ORF1AB was responsible for producing this particular protein, which is the protease enzyme. So we took the sequences from the NCBI viruses, and then we took all these sequences, and then we use an in-house PERT script to extract the desired protein from these all genomes, and then did a multiple sequence alignment to figure out how these sequences are similar or different from each other. And we found out that there were two set of sequences. One set there was no mutation with compared to the PDB structure which is available on the PDB, which was the 6LU7, and there was a set of sequence near about 40 sequence in that particular set that we had, which had valine mutation, which is valine deletion at 3 positions. So then we headed for the ligand preparation, for which we took a list of HIV inhibitors which we take from the drug bank, and then we designed their analogs, which are given on the screen, the number of analogs for every inhibitor set we generated, and then certain inhibitors from the SARS from literature we took. Then we took some peptidomimetics which was developed by my mentor for this process, and then we did an enzyme inhibitor docking using Autodock Vina 4 using these coordinates. So these coordinates were particularly selected because they had the active site within them, main residues which are responsible for this activity, the cysteine 145 and HIS 41. So we focused on that, So what I was mentioning before, that we can see there are valine deletion in near about 41 sequences, uh, where the 3 positions they have a deletion. So we were suspecting that this could change the activity. If this mutation could change the activity with reference to the inhibitor that we are designing. So we also checked the sequences specifically for the Indian region. The time that was I was doing the project, there was only 2 sequences available. So the sequence that was found in India, from origin from India, had no change from the structure that was available on the PDB. So, so I modeled the structure which has a valine deletion, and then superimposed it on the structure which was available on the PDB, and found out there was certain deviation. Uh, we can see at the histone 41 there was a certain deviation over here. So this could also lead to change in the activity with reference to the inhibitor. And then there was another side which was found during the blind docking, that it was also showing certain propensity towards the ligands that we are trying to bind it, the inhibitors we are trying to bind. So we performed the docking on the structures, and we found out that among the library of 2500 molecules, we found these molecules fit with particularly good binding energy, maximum reaching to minus 8.1, and with a good ADMET background, which is satisfying all Lipinski rule, and then with the certain criteria defining the druggability of the compound, like if they are druggable or not, like having a particular total surface polar area, or being a molecular weight below the 500, or is it soluble, is it can be absorbed by the gastrointestinal tract, do it permeate the blood-brain barrier, or is has some activity with the cytochromes. These activities were analyzed, and it was found that these compounds in the table were having a good activity as well as a good docking score. So here is a graph which represented, and we found this compound that 143934390, which is its CID from the webcam, was having a good activity against the protease. So it could be a good inhibitor. And so here I am trying to tell you how good it was binding. So you can see the ligand within the binding pocket, and here you can see there is a hydrogen bond which is making with the cysteine 145, and it has a very good binding when it comes to the surface to surface interaction. So it is ensuring that it has a particularly good interaction with the protease molecule in its active site. And so for that we tried to confirm it from the protein plus server, and we found the same interaction that we were having from the results of Autodoc Vina. So which was really assuring. So we can see here that it is showing a good interaction with cysteine 145, and here's the 3D structure depiction where you can see. So what we found that in the sets that we had, these molecules have a good binding energy, and they have a good ADMET score. Even after that, there are certain molecules that we had in our set, which are already marketed, also had a good binding energy and a good ADMET profile, so which could be repurposed for the COVID-19. Among them, the best performing was the stachinavir and the brekinavir. And the library of peptidomimetics that we designed, among that we had few molecules that we could use for COVID-19 protease inhibition, which are QSAR modeled and have a good ADMET profile. So what are we looking forward to? After screening the library of 2,432 molecules, we want to profile these top performing molecules for their molecular simulation. If we found that these are performing good in their molecular simulation, so we can push them for their synthesis as well as their drug trials in future. Thank you.
Thank you, Mr. Sharma, for your presentation. I think now we can move on to the last presentation of the day by Miss Navneet Kaur. Miss Kaur, are you there? Miss Navneet Kaur. Hello, Miss Navneet Kaur. Oh, she's there. Miss Kaur. Miss Kaur, please start your presentation.
Hello.
Hello, we can hear you. Please start your presentation. Okay, I think Miss Navneet Kaur is having. Oh no, you're. She's. Yes. Please start your presentation, Miss Kaur.
Sir, actually the screen can't be shared at the moment. I'm trying, sir.
Mm-hmm. Open your presentation first and then try to select the window.
Okay, sir. I'm doing. Hello.
Yeah, you are audible. Please open that presentation first and then share it.
Sir, I have opened the presentation. Is it visual?
We see only the file directory. I think Ms. Kaur, you are having problem in presenting your work. So should we postpone it for tomorrow? You can do presentation tomorrow.
Yes, sir. Yes, sir.
Okay. Okay, then the presentation will be tomorrow. This was the last presentation for the today. Now, I take this opportunity to thank all the organizers, particularly Dr. Vijay Tripathi and Dr. Pragati Mishra, for organizing this conference, international conference on recent advances in biotechnology, bioinformatics, and biochemistry, CONIAPS 26th chapter of this CONIAPS. I'm really grateful that they gave me the responsibility to introduce eminent speakers to the audience. I thank convener of this conference, Dr. Jonathan A. Lal, who is Dean of Jacob Institute of Biotechnology and Bioengineering. And not it cannot be over without thanking the head of the Department of Molecular and Cellular Engineering, Professor Kapil Lawrence, who was vital in organizing this conference, and obviously the university, Schwartz, who has played a big role in organizing this conference and extending all the facilities to organize this conference online in time. And without saying thank you to International Academy of Physical Sciences, Prayagraj, and Mahatma Gandhi University, Kottayam, Kerala, who are also involved in organizing this conference online. Thank you, good luck, and we'll meet tomorrow for the next session, though I'll not be there in that session. I'll be listening. I'll be an audience in that session. And tomorrow, again, though it is Sunday, but the presentation will start at 2 p.m. tomorrow. Thank you. Have a safe evening and good night. Bye.